as I am to begin the second day of insightful GIAN course on advances in experimental therapeutics. We have an engaging lineup of lectures and tutorials today by our esteemed speakers. Let's dive right into today's schedule. To begin with, we have Dr. Aman Bhalla, who will be delivering the lecture in our series titled Asymmetric Synthesis in Drug Design, Need and Basis. Dr. Bhalla has been guiding us through the crucial strategies of chirality and selectivity in drug development. We are eager to continue this journey with him. Let's welcome Dr. Bhalla with a round of applause.
A very good morning to all present. Okay. So yesterday we have started the asymmetric synthesis in drug and designing. That what is the need of the asymmetric synthesis. Okay. So we are continuing with that part, and as I said in our previous lecture, that this topic is selected so that you are able to get the hints, you are able to get that what type of synthesis you have to plan so that you get your desired compound in a high enantiomeric purity, so that the beneficial effects of particular isomers can be taken into the next steps. So as I said earlier, that if you have a one or the two enantiomer in your synthesis, you have to purify them, you have to characterize them, you have to give the data of both the enantiomer in the market, then it will be approved for further use. Okay. So we move to the next, which is the most important slide if you have to study about the asymmetric synthesis. Okay. That is the selective synthesis of enantiomers. Okay. If you see the reaction, which that is a very simple reaction, can you see what is in your substrate? It is a simple aromatic ring and the carbonyl functionality. It is not any complicated reaction scheme. You just see in your substrate it is a achiral. Okay. Now if you see the final products, you are getting the reduction of the carbonyl compound, you are getting the corresponding alcohol molecules. But if we are talking about our substrate, that is carbonyl compound, it is planar in nature, it has sp2 hybridization. So if you try to add any group on it, it has a possibility that that group can either attack from the upper plane or it can attack from the below plane. Okay. So in a simple reaction, if I show this is the carbonyl group and you are adding hydride ion on it, the hydride ion can attack from the upper plane or it can attack from the lower plane. That means you have a 50-50% chances. So I just quote a very simple example. We are sitting in this room. Okay. If 100 students are coming into this seminar room, we have two doors. 50 students can come from back side, 50 students can come from front side. This is the possibility which we can execute. But if we close either of the door, if we close the backward one or the front one, then what is the possibility? Only from the one side. Okay. So what is asymmetric synthesis? It is the synthesis in which a chiral unit, which is the ensemble of your substrate molecule. What is a chiral unit? If you see, there is only aromatic ring and the carbonyl group. So that is a chiral unit is converted into a chiral unit in a such a way that the possible stereoisomers are formed in unequal amounts. So this is your asymmetry. Okay. Now, if you see, you are getting two enantiomer, that is S or R, but what we have written below, that is non-racemic, unequal mixture. Okay. So, how you planned such type of synthesis where the possibility generally is 50-50, but you are forcing the reaction that this possibility should be unequal. If it is unequal, then your asymmetric synthesis is going to start. Okay. So, it is a desired proportion, that the enantiomer which you desired for your final target. What is your final target? You want an antibacterial molecule, antioxidant, anti-cancer, antiviral, whatever the property you are desiring. Okay. So, that is why either you get A or either you get B. Now, I am just asking, what is the difference between A and B? It is only the stereochemistry, the arrangement of hydrogen and OH in the three-dimensional space. In first picture, that is hydrogen is below the plane. In second molecule, the hydrogen is above the plane. So, this is the only difference, but you want such molecule in unequal amount. So, which enantiomer is more preferable as compared to the other one, that tell you the enantio selectivity. So, if you see yesterday lecture of Professor Narinder Singh, there are number of molecule which have a chiral unit. If you present any molecule in your studies and say that my molecule is in chiral, the first question of the reviewer is that what is the enantio selectivity and what is the diastereoselectivity. You cannot just put the chirality and you say that I have prepared it. Nobody is going to take this, you know, because yesterday we discussed the thalidomide case. Everybody wants that if it is a chiral molecule, you have to report the enantio selectivity or the diastereoselectivity of such molecule. So, that means you have to get the proportions, that which enantiomer is in majority and which enantiomer is in minority. So, if you plan a synthesis and the synthesis works, and if you are getting A and B in the 50-50 ratio, then you say your reaction giving racemic product, because both the enantiomers are in 50-50%. But if you see the percentage, your A is 90 and B is 10. That means how much is the excess of A with respect to B? That is 80. If A you get 90 and B you get 10, then what is the excess? 80. So, that is your 80% excess of one enantiomer over the other. But if you perform a reaction in which A is 100% and B is 0, okay, then your one enantiomer is formed in a 100%, other is out of this system. So, what you call, if you get only one enantiomer, that is your enantio specific reaction. But if you get the enantiomeric, if one is 90, other is 10, then you call it is a enantio selective reaction. So, when any chiral reaction is going to perform, you have to explain in such type of terminology. The reviewer or the judge or the interview panel expert, whenever you are presenting your work to them, they going to ask, is your reaction enantio specific or enantio selective? Then you have to say that if you get only one type of enantiomer, your reaction is enantio specific. But if the ratio is 90-10, 80-20, 70-30, then you say that my one of the enantiomer is in excess as compared to the other. So, that is enantio selective reaction. So in asymmetric synthesis, this terminology is required because whenever you are going to see the asymmetric aldol reaction, asymmetric Diels-Alder reaction, asymmetric Sharpless epoxidation, hydroxylation, every time the question comes, your reaction is whether enantiospecific and enantioselective. So I just ask one question to the audience, that if you desire your reaction, what will you desire? It should be enantioselective or enantiospecific? Enantiospecific, because you are getting only 100% of one type of enantiomers. Okay. So now till here you know that you need asymmetric synthesis, you need pure compound. Why you need? You have an answer, क्योंकि every isomer may be related to one type of particular activity. So our objective is clear now, that we required pure compound. Now the question is how we can require, what are the methods that which we have to see we can get the enantiomerically pure products. So if you see on your screen, these are general methods. Okay. So if you see, first is resolution of a race mate to chiral compounds with recycling of undesired enantiomer if possible. I will take one by one this reaction. Okay. Second is synthesis of chiral compounds by deriving the chirality from naturally occurring chiral compounds available in chiral pool. Third is microbial or enzymatic transformation, and fourth is catalytic asymmetric synthesis. These are the general methods which are available during your course of reaction. Now what is your course of reaction? You start at synthesis and you see in your substrate chiral moiety is present. You perform any reaction. What is any reaction? आपने asymmetric synthesis का पहला reaction क्या देखा था? You have a aldehyde group, carbonyl group. You are reducing it into alcohol. So this is your general reaction. You perform this reaction, and as per theoretical concept, you are getting 50-50 of both the enantiomer. As I said that two doors are available, anybody can come. Now I am giving the lecture, this door has been closed. Now whosoever is coming, he or she is coming from that side. Okay. So what is the meaning of this? One side is blocked, other side is open for the entry. But in start, both sides are open for the entry. So if any reaction you have started and by chance or by theoretical concept you are getting two enantiomers, then we don't have to worry. At least we are getting two enantiomeric products. What we have to do? We have to resolve them. So that is what? That is resolution of enantiomers. So resolution of enantiomers कैसे कर सकते हैं? What is the difference between enantiomer and diastereomers? We are saying that you cannot separate enantiomers, but we can separate the diastereomers. So if you are getting any enantiomer, see in your substrate, what is in substrate? Plus minus denote what? It is a racemic mixture. And what is your target? You want to get pure plus and pure minus, so that you can analyze what type of activity is available with such type of products. So generally what you done? Take the substrate, treat it with pure optically active reagent. That optically active reagent can be plus or minus anything, but that should be a pure enough. So what is the final product? आपके enantiomer में एक chiral unit है, आपने एक reagent में chiral unit लगा दी, तो क्या बन गया? Diastereomers. So you can see plus substrate A star minus substrate A star, that means you have a pair of diastereomers. Now you separate them. Diastereomers can be separated by fractional crystallization, distillation, there are different methods. So once you separate them, then you take out the reagent which you have introduced to make as a diastereomer. So you remove the A star, you get your pure plus and pure minus, so you can further use them for the different activity. So this is your resolution. Okay. Now if resolution is the only choice, we don't have to do the asymmetric synthesis, do the reaction, get the racemic mixture, separate them. But is it possible that we can separate them easily? So if I tell you, just give me one example, if you treat acid with alcohol, what is the final product? Ester. If you treat acid with amine, what is the final product? Amides. And if you get back that product, what you have to do? The hydrolysis of ester, hydrolysis of amide. So that means your complementary nature should be there. If you have a ionic salt, amides, ester, in such cases they should be resolved back to the starting substrate. My only query is, if your corresponding A star is not available, then how you are going to resolve it? In your substrate you have a positive and negative isomer, you have to resolve it, that means you have to react it with A star. But if corresponding A star or a reagent is not available, how you are going to resolve them? So if you have a star available which can react with your racemic mixture to prepare the diastereomer, then this method is good. Okay. So even if you get the racemic mixture, choose the appropriate A star, convert them into diastereomer, separate them, remove the A star, you will get your pure enantiomeric products. Okay. But if corresponding A star is not available, then this method cannot be used for the resolving of your racemic mixture. Then resolution cannot be used. Okay. So then we move to the another, that is your chiral pool. What is a chiral pool? Okay. I just want to explain you first reaction, then go to the theoretical concept. Now can you see your target product? That is your enantiomerically pure lactone. What is lactone? The ester unit in a cyclic form, that is your lactone molecule. Can you see ester unit in your final product? And you are able to see the chiral unit. What is the chiral unit? Hydrogen and COH. Hydrogen is your below the plane, COH is your above the plane. Now if I say that enantiomerically pure lactone unit is a part or a molecule which is required for your antiviral drug, then your aspect is that you have to prepare this unit. Okay. Now how this can be occur? See, in starting reaction, starting substrate, that is L-glutamic acid. In L-glutamic acid, can you see the chiral unit? Is it exactly the same which you desire in your product? You see hydrogen and COH, they are present on the same plane which you required in your final product. Okay. So you choose a substrate which have all already the desired chirality of your product. Now what you done in this case? You just convert the NH2 to corresponding OH, diazotization, and then you do the cyclization. What is cyclization? Acid react with alcohol to give you what? Esters. Okay. So in this case, if you do this cyclization of OH and COH, you get that cyclic product. Now can you tell me what the chirality which is remain in the substrate directly goes to the product? So what is that thing? That is called chiral pool. The nature has provided us number of molecule which have a chiral unit. Okay. So if such type of chiral unit you required in your product and they are already available in your chiral pool, then just plan a synthesis in a way that unit should be carried as such. Now can you see in this reaction the chiral unit which is present in the substrate that directly move to the product without changing anything. Okay. So what is this method? This is an appropriate chiral precursor from chiral pool can be modified in a series of chemical step to a desired chiral compound. So nature से ही आप एक ऐसा substrate, ऐसा precursor choose करिए जिसके पास आपकी required desired chirality हो. आप series और step of reaction perform करो उसी सबस्ट्रेट के साथ. पर क्या होना चाहिए? नो स्टीरियोजेनिक सेंटर रिक्वायर्ड इन द डिजायर्ड chiral कंपाउंड शुड बी इन्वॉल्वड इन केमिकल ट्रांसफॉर्मेशन. जो सबस्ट्रेट में chiral यूनिट है जिसको आप एज सच प्रोडक्ट में चाहते हो, दैट शुड नॉट बी डिस्टर्ब्ड. तो अगर आप इस रिएक्शन में देखो, स्टार्टिंग सबस्ट्रेट से प्रोडक्ट तक क्या हमने chiral यूनिट को टच किया? जैसे सबस्ट्रेट में थी वही हमारी डिजायरेबिलिटी थी, उसी को हम एज सच फाइनल प्रोडक्ट के लिए लेकर चले गए. ओके. सो दैट्स मीन द केमिकल स्टेप्स हैव बीन इन्वॉल्वड इन अ सच अ वे. व्हाई यू चूज मॉलिक्यूल फ्रॉम द chiral precursor? बिकॉज यू रिक्वायर्ड अ पर्टिकुलर टाइप ऑफ chiral यूनिट. So that chiral unit should not be disturbed at all. It should be directly moved to your final product. So no stereogenic center should be disturbed, no racemization should be allowed. ऐसा नहीं है कि आपने प्योर chiral कंपाउंड से शुरू किया, स्टेप ऑफ रिएक्शन ऐसे किए, जाके वहां पे फिर रेसमिक प्रोडक्ट बना दिया, फिर उसका क्या फायदा हुआ? फिर हमें उसको दोबारा से किस स्टेप में लेकर जाना पड़ेगा? रेजोल्यूशन. दैट्स मीन a synthesis should be planned in a way your chiral unit should not be disturbed at all. Only chemical transformation should be done. Okay. So however, stereogenic center may undergo inversion or retention, but chirality should not be lost. Retention and configuration हो सकती है. आपने SN2 and SN1 reaction पढ़ा होगा. Okay. If you perform a reaction on chirality in SN2, what is the configuration of the product? Inversion. But if you do in SN1, it is racemization. Okay. So then what is the limitation of this method? Only limited number of natural products are available. Only few functional groups are available. Now you start preparing the asymmetric synthesis, synthesis where you get the unequal amount of products. So you first start the method of resolution. अगर उससे पॉसिबिलिटी होगी ठीक है, उससे पॉसिबिलिटी कब नहीं होगी? जब आपके पास कॉम्प्लीमेंट्री ए स्टार अवेलेबल नहीं होगा. सो देन यू मूव टू द chiral pool. अच्छा भाई, हम chiral pool में देख लेते हैं अगर कोई सबस्ट्रेट मिल जाए जिसके पास हमारी डिजायर chirality हो. तो आपने चेक किया it is not necessary that always you get your desired chirality in the natural available products, they are very limited. Second, if you get the complementary groups are not present which has to be converted into the unit. अब आप इस reaction में दे you required OH and COH so that they can condense together to make a cyclic electron. अब आपके starting substrate में ये chiral unit तो present है लेकिन OH and COH ही नहीं present है, तो फिर cyclic molecule बनाएंगे कैसे? So that limitation can also be there. So we move to another method, but this is just a list that these chiral starting materials they have been already provided by nature. So it is called chiral pool. Okay. So the question comes, what is chiral pool? It is a list of molecules which has been already available in your nature, which has been extracted from different plants and sources, which can be used for the chemical synthesis of chiral products. So आपके पास amino acids है, amino alcohols है, hydroxy acid है, terpenes है, alkaloids है, carbohydrates है, these are units, they are already present. So you can pick them, you can convert into your target products. So that's mean chirality is already in your substrate. Okay. So we go for resolution, we go for chiral pool. Now we come another method which is enzymatic methods. अभी हमने पढ़ा था enzymes are stereospecific in nature. I will show you one picture which you will explain. First see number 1, number 2, number 3. Just see the substrate, you don't have to remember anything, but you have to just give a picture in your mind. In first case, you are doing the hydroxylation across carbon-carbon double bond. What is hydroxylation? You are adding OH group across the carbon-carbon double bond. Now, can you tell me what is the stereochemistry of carbon-carbon double bond? It is planar in nature, it is sp2. Okay. So if you are going to add 2 OH group on the 2 carbon of double bond, either the OH group can come from the above, either the group can come from below the plane. So this is your hydroxylation. Now can you see in this example both the OH group are on the upper side, above the plane. You are getting cis diol enantiomeric excess कितनी है इसकी 100%. That means this is a enantiospecific reaction. You see in second example, what is the difference? At both the carbon double bond क्या difference नजर आ रहा है आपको? सेम ही तो ग्रुप है, COH and H. नीचे कार्बन में आप देखो सेम ही ग्रुप है H and COH. But when you are getting the product, now can you see the addition of water? One side you are getting OH, other side you are getting hydrogen in the presence of some enzyme, that is fumarase. Okay. Now go to the third reaction. You are seeing 2 carbonyl group. One carbonyl group is part of your ester unit. Okay. Now you are seeing the target product, the enantiomeric excess, you are getting the reduction of 1 carbonyl group where you are getting the OH group. Now can you tell me this OH group is below the plane? If your carbonyl is in the plane, OH can come from the upside, OH can come from the below side. But in your product, you are getting the OH from the below side. So in these 3 reactions, you are getting enantioselective and enantiospecific products. Now see, if I am saying the cis diol is a molecule which is a synthone for the formation of amoxicillin. Say example, I am saying the product number second that is the synthone which is used for the formation of medicine dolo. Okay. The product number third is the synthone for the making of anti-cancer drug. So for all these preparations I required this in enantiomeric form. So what I have to do? I have to just choose the appropriate enzyme. Okay. So the impressive enantioselective of enzyme due to their capability of molecular recognition at the active site is most useful in this case. Okay. So we say enantiomers have same physical and chemical property, they have different biological property. Why? Because enantiomer are only differentiated with respect to their arrangement in the space, and enzyme is very specific. It can check that whether a particular group is above the plane or whether a particular group is below the plane. So you can see enzyme now offer an alternative for every organic reaction, providing a unique control on enantioselectivity. Okay, that's mean how this enantioselectivity can be best. I just show you one picture. Now can you see in this picture the portion which is shown in a blue color, that is your enzyme. Okay, this enzyme is used for the oxidation of one amino acid, that is L-alanine. Now can you recognize what is the meaning of L and D? This can differentiate the two enantiomers whether they are rotating on left side or right side. So this is the one enzyme which can oxidize the amino acid alanine. Now can you see in this case this enzyme is particular for one type of enantiomer, and that enantiomer is L-alanine. But if you see, you try to add the D-alanine, it is not fit. So that is your stereo specificity of a particular enzyme. Now if you see what is the difference, you are just shifting the hydrogen and NH3, but enzyme is not providing the space. It has a particular space for NH3, particular space for hydrogen. If you just drift the molecule, it is not going to fit that molecule. That is the best part of the enzyme. Okay, so they are going to provide only the space active site interaction for your L-alanine, not to the D-alanine. So that means they are very specific to hold only one enantiomer. Now if you see in this case, you are adding the lactic acid molecule. Now you can see enzyme has a 3 different sites where your lactic acid molecule is going to place. Now in first side, OH and COH group have the accurate position where they can fit, but on the other side if you change the stereochemistry, that is just the mismatch. OH can go to the big hole and it can come back. It is not going to held tightly with the enzyme. So if it is not going to held tightly with the enzyme, the reaction cannot be initiated. Okay. So what these examples are showing, why I have gathered this example, that in what is the difference in first and second substrate? The COH group is in first case present trans to each other, but in second case it is on the same side. So if you take the hydration of this molecule in the presence of fumarase as the enzyme, you get only product in first case, not in the case of second. So what all these example explain? This example explain that enzyme is the very good method to stereo specifically, enantio specifically to form a particular type of product. Now what is the lacuna in it? What is the issue with it? that you have to isolate the enzyme, you have to purify the enzyme, you have to identify the enzyme and then stabilize. You must have a data bank. If you record IR spectra and NMR spectra, how you check that your product has been formed? In IR you have some data that from this to this functional group, from this to this functional group. If you record the NMR spectra, you know if such type of coupling, it is doublet, it is triplet, it is quartet. So if you have to do such type of reaction, for example, you have to make cis diol and you have a benzene molecule with you, what you see? Just check what type of enzyme is available. If it is in your laboratory or you can purchase it, do the reaction. You get your cis diol in 100% enantiomer specific way. But the question is, this data bank should be there. But if it is not available, how you can do the asymmetric synthesis using enzymes? Okay. So enzymes are a very good method to do the asymmetric synthesis, but if data is not available, if your substrate has such type of functional group, with respect to that particular enzyme is not available, how you are going to use this method? Then there is a problem. Then you have to go for some alternative method because particular enzyme is not available. If it is available, go for it. You can get your enantiomer selective and enantiomer specific product. So till now we have discussed resolution is the one method, chiral pool is the second method, enzyme is the third method. If all these three methods are good, available, perform the asymmetric synthesis, do the reaction, get your pure products. But if all these three methods are not suitable, not available, then what is the plan? Plan has to be there, because why plan has to be there? It is not just a material which you are preparing. It is not just you prepare a reaction and you are using for any furniture. These are the molecules which has to be converted into the drugs, and that drugs are going to use by the mankind. So you cannot wait till here, okay, these three methods are available, if we are get our final products, okay, you have to go for some alternative. So for that alternative, the part which we are discussing, the part which we are talking is the asymmetric synthesis should be planned in a such a way that you are getting either A or either B in enantiomerically pure form. Okay. So this is the fourth method which we have to study about, the catalytic asymmetric synthesis. So till now this method, any questions with you? Is it understandable? My language is okay? फास्ट तो नहीं, स्लो तो नहीं हूँ? क्लियर हो रहा है आपको? Because I have done it the 10 to 15 year ना. This is only for you. If something you can get from it in your synthetic journey. Okay. So in all the previous methods have one or the other problem associated with. तो फर्स्ट मेथड में रेजोल्यूशन में क्या प्रॉब्लम थी? कैन यू, जो फर्स्ट मेथड है रेजोल्यूशन का, उसमें क्या इशू आ सकता है? एनीथिंग यू कैन ऐड? उसमें आपको कॉम्प्लीमेंट्री ए स्टार नहीं मिलेगा तो डायस्टीरियोमर बना सकते हो? यू कैन नॉट मेक. सेकेंड मेथड में हमें क्या प्रॉब्लम है? कायरल पूल में हमें क्या इशू आ सकता है जतिन? जो सेकेंड मेथड है हमारा कायरल पूल वाला, उसमें क्या हमारा ड्रॉबैक है? जो आपकी डिजायर्ड कायरलिटी है प्रोडक्ट की, वो कायरल पूल में अवेलेबल ही नहीं है. थर्ड मेथड है एंजाइम, इसमें क्या इशू है, जो रिक्वायर्ड एंजाइम चाहिए वो आपके पास उसका डाटा ही नहीं है। देखो दिस इज नॉट अ वेरी बिग साइंस, आफ्टर स्पेंडिंग यूर 1 hour here, if you are not even able to answer in a one keyword, then what is our gaining? Okay, I am not saying that you have to study asymmetric synthesis, what type of research you are doing, it's the best one. Okay, but if we are spending a 1 hour here, any keyword which should be with us, okay, for the general knowledge, that is going to help in a some way. Okay, so it is the simply we are saying previous 3 methods have one problem, जनरल वे, इसमें बहुत बड़ी साइंस तो है नहीं। सर रेसमिक मिक्सचर बनाया है, नहीं सेपरेट हो रहा है, दिस विल बी द फर्स्ट प्रॉब्लम। रेजोल्यूशन इज व्हाट, यू हैव अ रेसमिक मिक्सचर, यू हैव टू सेपरेट देम द एनैंटियोमर। सेकेंड मेथड आपने किया कायरल पूल। कायरल पूल क्या होता है, नेचर ने ही कोई ऐसा मॉलिक्यूल दे दिया जिसमें कायरलिटी है। आप सब रिएक्शन तो लगाते हो ना लेबोरेटरी में, कोई ना कोई सबस्ट्रेट तो चूज करते हो, तो उस सबस्ट्रेट में अगर आपकी डिजायर कायरलिटी नहीं है, दैट्स मीन वो कायरल पूल में अवेलेबल नहीं है, मैं कैसे सिंथेसिस प्लान करूंगा। ओके, थर्ड मेथड आपका एंजाइम आया, आपने देखा ये मेरा सबस्ट्रेट है, मुझे ये फाइनल प्रोडक्ट चाहिए, उसका पर्टिकुलर एंजाइम चेक किया, वो एंजाइम अवेलेबल ही नहीं हुआ, तो वो आपका ड्रॉबैक हो गया। सो यू मूव टू समथिंग वेयर यू हैव टू गेट योर फाइनल प्रोडक्ट। ओके, सो दैट इज हाउएवर the last and the most important alternative is to begin with a achiral compound. Your starting substrate is achiral. To generate a single enantiomer of chiral desired compound by chemical asymmetric synthesis. अब आप देखिए यहाँ पे आपने क्या रिएजेंट ऐड किया है, मैंने एग्जाम्पल वो लिए हैं। इफ यू सी yesterday lectures, Dr. Ninder, there are complex molecules. You are see the big type of publications you can get where you are talking about the complex molecules. I have just taken a very simple examples where you can understand the substrates and the product. Now if I am saying see your substrate, there is one aromatic ring and one carbonyl group, and what you are going to do, you are going to add H negative. H negative कहाँ add होगा, आपके पास कार्बोनाइल ग्रुप है विच इज पोलर इन नेचर, बट इन मीन पोलर, यू कैन क्रिएट पोल्स, कार्बन पॉजिटिव ऑक्सीजन नेगेटिव बिकॉज ऑफ द इलेक्ट्रोनेगेटिविटी कंसेप्ट. So if you create a polarity, then you can add H negative there. So your H negative is going to add to the carbon, you get O negative and the system will give you H positive, you will get the reduction of your starting substrate. But if you do the reduction, the possibility, what is the first possibility? Your H negative is going to the carbonyl carbon. अब यहाँ से आपकी प्रॉब्लम भी स्टार्ट होती है, यहाँ से आपका फाइनल फ्रूट भी स्टार्ट होता है, क्योंकि यहाँ पॉसिबिलिटी है. When H negative is going to the carbonyl carbon, it has the two possibility. Either it can go from the above side and it can go from the below side. So the desirability is or. Desirability is either you want to get A or either you want to get B for your particular objective. Okay, so that is the important part of your asymmetric synthesis. Why it is important? Now see, this is just a simple reaction, aldol reaction. अब aldol reaction तो हमने +2 में किया हुआ है, नेम सबको याद है कभी? Why it is called aldol? In this its product you have a aldehyde unit also and OH unit also. Now if you see, you are going to add, you are going to do a aldol reaction, you are going to add a enolate. See the product number 5. आपने प्रोडक्ट देखना है और उसमें मैं जस्ट आपको एक पॉइंट बताऊंगा, उसको आइडेंटिफाई करो. पॉइंट नंबर 2 एंड पॉइंट नंबर 3. अगर आप अपने प्रोडक्ट में कार्बन सेकेंड एंड कार्बन 3 को देखो तो उसमें कौन सा ग्रुप अटैच्ड है? R and OH. Now if I say this R and OH in 3 dimensional कैसे प्रेजेंट होगा? Can you see the pictures below? Relative stereochemistry with respect to carbon 2 and with respect to carbon 3. बड़ा सिंपल सा क्वेश्चन है ये. कार्बन सेकंड पे R ग्रुप लगा हुआ है, कार्बन थर्ड पे OH ग्रुप लगा हुआ है. जरा R और OH की 3 dimensional स्पेस में अरेंजमेंट विजुलाइज करो. सो इफ यू आर visualizing the arrangement, what will be the possibility? Can you see in number 1 R and OH on the same side and they are on above the plane. Okay, R and OH on the same side, they are on above the plane. Now go to number 2, R and OH still on the same side but below the plane. Okay, now move to picture number 3. R and OH, they are opposite to each other. In number 3, OH is above the plane, R is below the plane. Now move to number 4. R and OH, they are opposite to each other, but OH is below the plane, R is above. Now if you are doing the aldol condensation reaction with the chiral unit, you are still no मेरा aldol product बनेगा, but if you see the possibility are 1, 2, 3, 4. Now here comes the important point. Why aldol reaction is very synthetically important? आपके एल्डोल में एल्डिहाइड भी है और अल्कोहल भी है. अगर आपके पास एल्डिहाइड है तो एल्डिहाइड ग्रुप के सारे रिएक्शन पॉसिबल हो सकते हैं. अगर OH ग्रुप है तो OH की सारे केमिकल रिएक्शन हो सकते हैं. So why these substrate are synthetically important? Because if your substrate has a multifunctional units, you can convert them into your chiral drug molecule very easily. Okay, so now you see the picture relative to 3 stereochemistry, 4 enantiomer are possible. Now I give you one assignment that after doing your PhD or MSc program, there are 4 different companies. There is a Sun Pharma, Cipla, Panacea Biotech. Okay, all these company, for Sun Pharma is using number 1 molecule for making drug. Okay, Cipla is using number 2. Okay, and your Panacea Biotech is using number 3. Okay, and Cadila is using number 4 as their substrate. Now from chemistry department you go and you join the company. They said you, any one of you join Cipla, Sun Pharma, Cadila, or they say I want only 1. Other person say I want only 2. Third one is say I want only 3. Now if you do this reaction, you are going to get 4 different product, 25% yield. So they can, this is not cost effective. आप एक सबस्ट्रेट से 4 प्रोडक्ट बना के दे रहे हो, 75% तो मेरा लॉस हो रहा है. Your assignment is that give me only 1 pure product out of these 4. Okay, so how you are going to plan this? Because if you are talking a simple reaction, you are adding only carbon 2 and carbon 3 with respect to each other, but that addition is going to give you 4 different products. Then how we are going to plan this type of synthesis? Then you have to plan, you have to study the asymmetric synthesis. How you can desirably get only number 3 आपने रिएक्शन परफॉर्म किया, आपके चारों प्रोडक्ट आ गए, आपने इन चारों की कॉलम क्रोमेटोग्राफी की, आपने इनकी प्योरिफिकेशन की, उसके बाद एक जो था आइसोमर उसको प्योरीफाई करके उसको आपने दिया, फिर उसमें इम्प्योरिटी रह गई, सेकंड की रह गई, थर्ड की रह गई, फोर्थ की रह गई, बैच फेल हो गया। देन उन्होंने कहना हाउ एफिशिएंटली यू आर, that we are desiring only one and you are giving us a racemic mixture, even 0.001% other enantiomer is present, that drug is not going to accepted by the drug authority of India and any other country. Why? Because we have already studied presence of another enantiomer can lead to what? So they want only pure enantiomer. So that's why you have to study the method, the generations where you can desirably get only one out of it. Okay, so any reaction which is going to give you a series of another product, I have chosen the very simple reaction. Now you have studied the Diels-Alder reaction. Diels-Alder reaction में क्या होता है? There is a one diene and one dienophile. If you see on your screen, this side is your diene and other is dienophile. Diene has two double bond has electron density, it can be used to make bond. Dienophile is single double bond. Okay, now see in this method, in Diels-Alder reaction, what type of product you are getting? A cyclic 6-membered ring with one double bond. Can you see your final products? Cyclic 6-membered ring with one double bond. Now what is the difference? You have to see what type of group you have placed. In diene you have added R, in dienophile you have added Z. Now आपने Diels-Alder reaction किया. Diels-Alder reaction is a very common concerted reaction. सिंगल स्टेप में आपके डाइन एंड डाइनोफाइल को ऐड किया, 6 मेंबर रिंग बन गई, मॉलिक्यूल हो गया. नाउ सी इफ योर डाइन एंड डाइनोफाइल हैव सम सब्स्टिट्यूएंट्स, व्हाट इज द अरेंजमेंट ऑफ दैट सब्स्टिट्यूएंट? कैन यू सी इन योर फाइनल प्रोडक्ट व्हाट यू हैव टू सी ओनली आर एंड जेड. आप इन 4 प्रोडक्ट्स को देखिए और R एंड Z की स्टीरियो केमिस्ट्री एक दूसरे से कोरिलेट करके देखिए. फर्स्ट इफ यू सी R is on the lower face, Z is on the upper face. In second case R and Z both are on the upper face. In down R is on upper face, Z is on lower face. In last case both R and Z on the lower face. सो जब तक तो आप मॉलिक्यूल को कॉपीज पे ड्रॉ कर रहे हो, दे आर 2 dimensional molecules, but when you are seeing the molecule in 3 dimension, you have to see in 3 dimension. Okay, in that way what is the possibility? Still the possibility is 4 different molecule. So now comes the concept that how this can be done. आपको रेजोल्यूशन वाला मेथड पता चल गया, आपको कायरल पूल वाला मेथड पता चल गया, आपको एंजाइम वाला मेथड पता चल गया. नाउ वी हैव टू से कि अगर ये तीनों मेथड ना हो तो हम अपना पर्टिकुलर प्रोडक्ट कैसे अटेन कर सकते हैं. ओके, सो दैट पर्टिकुलर प्रोडक्ट, for that we have to study about 4 different generations. Okay, they are the most important concept of asymmetric synthesis. आपको वो चारों जनरेशन का सिंगल स्लाइड में मैं आपको एक्सप्लेन करूंगा इन नेक्स्ट क्लास. वो क्लास जो है वो आपने मिस नहीं करनी, अदरवाइज एसिमेट्रिक सिंथेसिस का कुछ नहीं आपको आ सकता. बिकॉज हाउ asymmetric synthesis can be planned in your reaction. The question comes कि सर हम तो नॉर्मल सिंथेसिस कर रहे हैं, हम क्या कर सकते हैं? अगर आपकी नॉर्मल सिंथेसिस में कारेलिटी आ गई और आप प्योर मॉलिक्यूल दे पाए, एनी इंडस्ट्री विल पिक यू लाइक दिस। आप किसी भी फार्मा इंडस्ट्री के केमिकल सिंथेसिस ब्रांच में मूव करिए, इफ यू हैव अ एबल और अ कैपेसिटी टू जस्ट purify the compound, give them a exactly pure compound, they will immediately hire you. That if any knowledge is with you, even you don't perform this reaction, because here in your laboratory, whatever the synthetic plan we are in the laboratory or your research proposal, you will carry that. But when you move to any professional world, you cannot say that I am working on metal, I am working on beta lactam chemistry, I am working on transition metals or silica chemistry. No, whatever the plan they are executing, you have to fit into that. For fitting into that, whatever the excellence is with you, you are able to enter immediately. Even you have a knowledge, they will say okay, he or she know the concept, we will just put them into that concept. So इसके लिए हम करेंगे basis of asymmetric synthesis and generation. Okay, so that I guess we move into the next lecture. Any question from this portion? Anything you want to check? Is it understandable till now? My journey is that I have to explain you what is the need of asymmetric synthesis, what are the methods for the asymmetric synthesis. I guess you have some information with you. Okay, any question from your side? Manrit, clear hai? Have you studied asymmetric synthesis in your post graduation? यूजीसी का तो आई थिंक ये सिलेबाई का पार्ट भी है asymmetric synthesis. इसको करते हो कि स्किप कर जाते हो ज़्यादातर? So I just show you that why I am talking about the generation, like this is the part for generation where we have to study if stereochemistry at substrate. In second generation we have to study auxiliary, third we have to study reagent, and fourth we have to study catalyst. In 2021 Nobel Prize for the fourth generation, इस जनरेशन में नया कैटलिस्ट डेवलप हुआ, दैट इज द मेजर पार्ट ऑफ. अच्छा, काइनेटिक्स की नॉलेज है थोड़ी आपको? काइनेटिक्स पढ़ा हुआ है? काइनेटिक्स में अगर मैं कहूँ एज अ केमिस्ट, this is not related to any lecture or your work, if we have to say about any chemical reaction, how a chemical reaction occur, what are the parameters which we have to take? आपके पास एक substrate होता है, you have to convert into product, this substrate goes to the product via something, this via is question mark. This via can be your transition state, this via can be your intermediate. अब ये जो ट्रांजिशन स्टेट है, ये जो इंटरमीडिएट है, इसके एनर्जी पैरामीटर्स क्या है? सो आप क्या बोलते हैं, अगर एक्टिवेशन एनर्जी लो हो तो रिएक्शन की फिजिबिलिटी क्या होती है? ये जो ट्रांजिशन स्टेट है और इंटरमीडिएट है, इसको आप हमेशा अपर साइड क्यों दिखाते हो? इफ यू मेक अ एनर्जी ग्राफ, if this is the energy of your substrate, you try to show the energy of product this way, you make a graph like this. तो ये जो इंटरमीडिएट है, ट्रांजिशन स्टेट है, इसका ग्राफ में ऊपर एनर्जी क्यों होती है? बिकॉज दिस इज रीअरेंजिंग. रीअरेंजिंग मीन्स ये अभी ट्रांजिशन स्टेट में है, इसमें आपके ट्रू बॉन्डिंग अभी नहीं हुई है कि वेदर इट इज गोइंग टू कन्वर्ट इनटू योर 4 मेंबर्ड 5 मेंबर्ड प्रोडक्ट्स. ओके, सो इन दैट केस इट इज अ वेरी अनस्टेबल वन. स्टेरिक क्राउडिंग है, स्टेरिक हिंड्रेंस है, सो इसकी एनर्जी अप होती है. Once it come to the proper orientation, proper position, it can come down to your stable product. बस अब यही हमने बेसिस के लिए याद रखना है कि when from starting substrate to product we are going via something, how we control that something, because if you control that something to lower energy, your efforts will be low. अगर एनर्जी ज़्यादा होगी तो आपको एफर्ट्स कितने करने, आप रिएक्शन परफॉर्म करते हो. अगर नॉर्मल स्टरिंग कर रहे हो रिएक्शन कैन बी इजीली अकर, बट अगर आपका कंडीशन ऐसी है यू हैव टू गिव अ रिफ्लक्स टेम्परेचर. व्हाट इज रिफ्लक्स टेम्परेचर? यू आर प्रोवाइडिंग एनर्जी. तो व्हाट विल बी द बेनिफिट पार्ट, दैट हाउ यू मॉनिटर दिस in between part so that your reaction can be fast, feasible, easily enantioselective and enantiospecific. So that is your basis of asymmetric synthesis. Okay, so next lecture what we are going to do, two important aspect: basis of asymmetric synthesis and generation of asymmetric synthesis. Okay, thank you very much. Thank you. We can wait for some 5 minutes. हम वेट करें.
Thank you, sir, for that insightful lecture. We'll resume the next lecture in 5 minutes. Now, we welcome Professor Sanjay V. Malhotra from Oregon Health and Science University for lecture titled Drug Development and Pharmacokinetics Part 1. He will guide us through the fundamentals of pharmacokinetics, absorption, distribution and elimination, which are central for evaluating the success of any therapeutic agent. Let us welcome Professor Malhotra.
Good morning, Tanya. Good morning, everyone. Was it a heavy load yesterday? Okay, so I will talk about proteomics, but so the way I have designed this lecture is just to, even before going to proteomics, you first need to understand how biological space is unrolled, how it's investigated, how it's studied. I'm just trying to make sure my computer is silence. Okay. Why is not showing up there? This is connected. अंदर है, कहाँ की है? Oh, there was something there. All right, thank you. And I'll do that. Thanks. Okay, so Tanya mentioned I'll talk you about proteomics. I will come to that, but I will come to that in probably subsequent lectures on talking about the specifically the ADME, adsorption, desorption, metabolism, excretion, and toxicology. But that will be tomorrow. Before that, you need to understand how we understand the biological space. कल हमने बात की थी the chemical space. Just touched upon the chemical space or chemical solutions. Why chemical solutions are important? Because okay, the gentleman is missing. हम बात कर रहे थे कि these are all chemicals. Why we call biology. So basically now probably we are going to do okay. Even before we dive deeper into understanding, about the more of the disease biology, we need to understand what are the tools. औजार क्या हैं जिससे कि अपनी कार को खोला जाए, अपनी मशीन को खोला जाए, right? So we talked about chemical solutions yesterday, and today I'm going to touch upon how do we understand the biological space. So you saw this at the end of our lecture yesterday, how we have the various levels of organization that are needed to study, starting again from atom. Understanding of the fundamentals helps us to understanding the more macromolecules on the right side, and these are just examples of how biopolymers, proteins, nucleic acid, carbohydrates, and these how they are the building blocks of things that we see inside the cell. And then I shared with you this slide, for example, how these various different biomolecules with the protein, lipids, carbohydrate, vitamins, how they constitute different parts within the cell, how they are important in various functions of the cell, in formation of tissue, organs, and the entire body movement. Going back to, again, fundamentals, we need to understand. If you understand the fundamentals, this is the chemical side of the fundamentals. The chemical part that more we understand, better we have an understanding of the chemical space, it becomes easier to understand the biological space and investigating the biological space. We talked yesterday about, various, again, the changes, the differences that you see in various parts within the body. This is just showing one example and showing only the pH, for example, the acidity or the basicity within various parts, and the food that we eat or the things that we consume, how they are related and how they could be. It will be helpful in understanding what those things are, what their physical chemical properties, and in this case only, their acidity, basicity, or how or whether they are ionic or non-ionic, and understanding that basically will be helpful to us to unroll, to एग्जाम्पल यूज यूजली इसके जो ऑनियन हैं, ऑनियन की परतें एक के बाद एक आप हटाएं तो नीचे पता चलता है क्या है. So this is again in terms of the biological space understanding. The more we understand the fundamentals, we can unroll the biological space and we try to understand the biological space किस तरह से है. Okay, so after all, we are talking about, this whole focus system is the biological space. but how the biology is influenced by various factors. Khana, diet is one part of it, but if we look at ecosystem, from the cells, how the information, we talked only about, is there a pointer here? I'll use, you'll miss otherwise, I'll use the, okay. So, for example, we talked about cell, right? The complexity of the cell, but the environment and many other factors, they contribute the human health. And human health, that's cells is the physical part. The other factors, for example, the ecosystem that you live in. Remember yesterday in one of the things that I was trying to tell you that the things which are taken into account when you develop is whether the big data, as I was trying to explain to you, what big data does is that if you have, for example, 1 million people, 10 lakh अगर लोग हैं, you can break it down to what their age group is, what their gender is, what their eating habits are, what, you know, their lifestyle is, all of that, because all those factors impact what happens within the inside the body. अब आप सोच के देखिए, अगर स्ट्रेस है, अगर स्ट्रेस होती है तो स्ट्रेस क्या है? It is emotion that we express, but physically it is impacting something inside the human body. आपका बीपी ऊपर नीचे क्यों होता है? Something is changing, and that something inside the body is actually the biological impact it has. So the time that what I'm trying to convey is that there are many other factors that play role in how the biology is affected. So if you look on the side, on the right side, for example, our focus as human health is to understand all those factors and how those factors influence the biology, how they impact. And so on the right side, for example, if you see the human body, as I was telling yesterday, it's the most complex machinery, chemical plant, biological plant, whatever you call it, ever created by nature. So to understand that, to unravel that, to investigate what happens inside, we need to know. We talked yesterday about few tools about the chemical side, but as you see on the right hand side, we have to understand many how the environment affects, and that environment affects the body that leads to disease and the outcome of the health, right? And then in order to address that, we need to create therapies, we need to create उपचार कि उनका कुछ हो सके. So how do we enroll the biology? To understand, how do we understand from gene to drug? So in order to understand, as you start from, there is a disease, before it gets to the clinical trial, clinical health, we have to understand from this part what is happening at this level. That leads to what is the target, as I was telling yesterday, and then unrolling the target. So what I'm going to focus is how this part is done. How this part is done in order to get to this side, and then again we'll come on this side to see that. But how this part is done, basically how we understand from here to here and what are the techniques. So in this, probably in the next 30-40 minutes, I'll just try to show you, and then in the subsequent part I will talk more about the proteomics, about the omics in general, how the latest technology and is used and helping in trying to better understand the human body at the gene and the protein level. So this is, as you will see in my slides, the examples mostly that I will use are related to would be focused on cancer, again being one of the most investigated disease, the most complex disease, and the studies of cancer, the research on campus actually has led to many beautiful outcomes in terms of our understanding of biology, because in order to understand the most complex part, you try to create solutions that will help you to understand more complex part. So our understanding the biology has increased significantly because of that. So okay, so if you look at this slide, what I have is, from disease to normal, how do we bring that health in between to the normal side. In order to do that, we have to understand, and we have to be able to understand what at the gene level, for example, understand at the what the chemistry or the biochemistry at the cell level, physiology. So gene or protein is, in that reference, is the micro, smallest part. That how that affects the whole biochemistry interconnection between genes, how does it affects at the cell level, because see, gene is inside the cell. So if you understand that lower part, then you grow that to at the cell level. And physiology, how is the organ, in the organ, how the effect happens. So from gene to cell to organ, that leads to the clinical to the health part. So to understand what all of these things, we have to understand at the protein level, at the metabolite level, when we have at the gene part. And how do we do that? The information that is needed is what is happening at any of these, at the gene level. And I'll try to explain you the difference between the genome and the proteome. At the gene level, at the protein level, at the transcript level, which is the RNA part, what happens, and then metabolite, for example, how they break down. You take the food, for example, we talked yesterday, or you take the drug, how does it break down inside the body, and the broken parts, the metabolites that are created, how they are effective or not. Again, chemistry, all related to chemical part. But we need to understand how. So how do we understand this part? And then biomarkers. So I'll come to that, what are biomarkers, but something that you can follow, you understand, which is related to a gene, or if you're question of biological question of interest. So the biological system, at the molecular layer, subcellular, at the organ level, cell, tissue, organ, organism level. So how do we understand this? So the methods used, and you heard about the cell biology, molecular biology at the molecular level. So which is basically understanding of various different sub-disciplines. So we talked about the gene at the function level, at the function level. Then this, again, understanding the gene at and then you need the molecular biology, from functions to genes, genes to protein. If your organ is functioning, how genes are, or what genes roles are, what proteins roles are, right? And then bring back to what they influence the function. So that is basically molecular biology in a very simplified version is what it does. So if I expand it even a little bit further, so if you look at here, I have given very simple, and by the way, you will have these slides. You will have these slides, and then as I showed you yesterday, this morning I was trying to, Manbir, and I will give it Dr. Singh, and we were just talking, I'll have about 25 publications, which are not what I'm talking, but they are related to, so that will help you beyond what you listen to lecture. And you will also have, and Dr. Singh will have the access to all the slides that I'm showing here. So in your leisurely time, you can look at more in more details, and you can cross-reference with the other things that you want to use from the literature. So in a simplified way, if you look at the molecular biology, is the study of the essential molecules inside the biological system. So molecular biology is not just only related to human. I mean, if you are studying animals or plants, or there is a molecular biology at the molecular level. So it is, of course, associated with the gene level, is how things are structured, how tissues are structured, how organs are structured. Again, understanding, you use the molecular biology, how they function, how they regulate, what pathways are involved. Because I do not know, and I think in one of my slides in some of the lectures, I have drawn very simplified signaling pathways, because inside the cell there are proteins which are sitting on top of each other, or which are influencing connectivity of those. So as best as we understand, this is what we call the signaling pathway, how the signal is transmitted, or the information is moving from one to another, many different ways. So that's what is the term used as signaling pathways for this. And the processes that are required. So the processes that are required to reliably pass the genetic information is coded, as you see, from the DNA in the genes, the information is, how that information is passed from genes to generation of another DNA and RNA. Have you ever thought about how when cell replicate, how information is passed? अगर आप बीज टमाटर का लगाते हैं उसमें से रोज़ क्यों नहीं निकलता? रोज़ तो दूर की बात है, आलू क्यों नहीं निकलता है टमाटर के बीज में से? That information is stored inside and it replicates, right? Same thing. So that information is stored somewhere inside the cell, right? How do we understand that? What that information is? How do we understand how the cell is formed? So molecular biology is basically, those are the techniques which are used in order to understand that. I'm sure you at some point, I think you have seen enough of a life. ये विचार जरूर आता होगा कि हमने कहावतें तो बनी हैं कि क्या बोया पेड़ बबूल का तो कहाँ से क्या होए, you know that, right? So क्यों ऐसा होता है? At the biology level, why does that happen? Again, ask a question. अगर आपने एक बीज, आप देखिए बीज आप जर्नी आप कितनी करते हैं? Because I do sometimes, I like to go to a nursery and buy some seeds grow करने के लिए in a personal garden. But why is that there's some seeds they look alike? एक जगह है मैरीगोल्ड गेंदा का फूल और दूसरी जगह रोज़ निकलता है. आप सीड्स देखिए, कई सीड्स आपस में मिलते जुलते हैं. Why does it happen? Why is that information not one seed? एक दिखने में तो एक सा है, वो क्यों नहीं निकालता? So that information is stored. How that information is passed to understand, to unroll, is what molecular biology helps to understand that. So basically the goal is to understand, the goal of the molecular biology is to understand the biology, find genes and proteins and functions, see what is causing the phenotypic functions that you see, and see and understand that by using the techniques of molecular biology to understand when a gene or a protein is expressed in or not, because that information will help you to understand कि diagnose करना है कुछ तो किस तरह से diagnose करना है, उसके लिए कुछ treatment करनी है, उसके लिए कुछ modification, prevention करनी है, intervention किसी भी तरह की अगर करनी है, you need to understand. So basically these are, these questions can help, and that's what the techniques of the molecular biology help. So in next few slides, I'm not going to go into details because that's, you're all chemistry majors, so this probably be too much of the biochemistry, molecular biology techniques, but I'll just touch upon the things which are used in order to understand the techniques which are used. Similarly, सोचिए अगर आपने कुछ analyze करना, you use chromatography NMR to analyze the structure, right? And if you so same way, what are the techniques which are so you heard PCR, right? So what is that basically? It's a small scale. You can do experiments and just simply denotes how the design, the technique in order to understand from what happens when you are trying to take a tissue, take a cell, and at the gene level how you break it down. So basically this polymerase chain reaction is a small scale from 1 to 10 gene proteins. You can do PCR, restricted enzymes, you can do cloning, hybridization, and if there's a large scale, you also can go to the high throughput part. So basically you use polymer chain reaction, is one of the techniques which is used. The gel electrophoresis, which you has anyone of you done this, or have people done this? Okay. Do you want to share how you did this? You can take my mic. It's fine. I want to make it more interactive. So there is another reason. जब वो बोल रही है मैं देखूंगा ना कौन कौन सो रहा है. I'm just kidding. Don't take it seriously, okay? Go ahead. Go ahead.
So, like in the previous slide he mentioned about PCR, which is basically you're multiplying the DNA. Now you want to check that if DNA is available in the sample
No,
lab.
tell them, I know, you just look at them and tell them.
Now you want to check that if the DNA has been multiplied and it's available in the sample, so then you would run a gel. So basically the next slide that he showed was a DNA gel electrophoresis, and this gel is a matrix, and you load your sample in it, and there's a DNA ladder on the side.
Okay, can I ask you one thing?
Yeah.
वो लोड करने का ना तो सारा ऊपर से निकल जाएगा. Just tell what exactly the experimental design. How do you do that? So you take your gel.
Yeah. So you basically make a agarose gel, and so they're like 1, 2, 3, 4, 5, 6. These are different columns, and in these columns you will take your sample, like a few microliters of sample, and you will load it in that particular column, and then you would pass electricity, and DNA has a negative charge, so it will go down. Smaller the size, the more distance it would cover, and there's a DNA ladder on the other side, which is basically, we know the size value of these quantities on the first column, so then you can compare it and see what's the size of DNA that you collected. This is
Any question for her? मुझसे नहीं, उससे पूछना, उसने समझाया है.
I hope I've
Okay, so this is basically, as I was saying, I mean, if you have, how do you understand protein or उस level पे क्या हो रहा है, macromolecule, biological molecule, small molecule, yeah, please. So small molecule as you analyze, right? हमें small molecule को पता करना है, तो what do? We just go chromatography, उसका mass spec check किया, NMR check किया, उसके bonds connect किया, NMR ने बता दिया, IR ने बता दिया कौन-कौन से उसमें functional groups हैं. Those are the chemists, as a chemist we use. But biological molecules, so this is one of the techniques which is used. आपको अगर आपके पास में, if you have what genes are involved, in order to understand you have to have techniques. So this is one of the techniques which is there. And this is, you can, for now enzymes. So same way, you can have at the enzyme level, you can use the restriction enzymes, another technique which recognizes a very specific 4 to 8 base pair. So DNA is large, right? You break it down, and then you break it down to small pieces, and then you need other techniques. As she was saying, as the DNA breaks down, and again, as the smaller the molecule, it travels faster. Same way, if there are 4 to 8 base pair, there is another, this is reaction enzymes that are used. So as you see here, for example, for the enzyme in a microorganism, and these are E. coli, you probably I'm sure have heard of E. coli, if not the other two. So in there, when you look at, using these recognition and restriction enzymes, when you do this into 4 to 8 base pair, you can identify what it breaks down to, and then you can map it back in order to see what enzymes probably are present there, because you have to investigate to show what might be going on. So again, you collect these restriction enzymes, you collect, you do the cloning if you have to reproduce. Clone karna hai, clone karne ke baad mein there are many things that you want to study, so you want to clone those enzymes or those parts of the enzymes. But these are various, for example, there is a restriction fragment length polymerism. It's a technique which is used, again similarly, to identify the pieces, the fragments, of the larger molecule. And in doing so, you can actually clone and you can put pieces together, you can create vectors. So in my lab, for example, if we have to create an assay, so there's a specific gene specific assay that is to be created, you create a vector. You can put the sequencing and pieces together, but this is exactly what you need. And in order to now, for the technique, because if you create an assay, that assay has to give you a signal, right? Yesterday, as we were talking about, if the cells are, for example, if you take a small molecule, you test for any type of cell, let's say head and neck cancer, or whichever part. I'm just using, again, that's the example. At that end, so you have to create specific cell line by putting a vector, creating the pieces together. Now when you test your drug or a unknown compound, it has to give you a signal. कोई technique होनी चाहिए ना उसको करने के लिए, काम कर रहा है या नहीं कर रहा. तो how do you create those techniques? So by doing so, you create vectors, and in order to create the vector, you can put pieces together. आगे पीछे क्या लगाना है? At the end, you can put, for example, this luminescent fluorescent tags. So when your compound, your drug is actually is inhibiting or activating based on how you have created, it will give you a signal, and that signal is basically electronically, it is recorded. That's what happens. So that's what basically methods are. Behind the scene, they are very simple methods. So cloning and all, as you saw the pieces, breaking down the DNA to small pieces, and you can put it back together to create your own sequence that you want, and that sequence then you can use. So for example, why I'm telling you this, for example, as I was telling yesterday, and now you will see the slides. I have tried to give some thoughts in my each lecture, so how I can make the connection. So yesterday I showed you one example of the non-small cell lung cancer, right? A study that showed out 10,000 lung cancer patients, and when they studied, it showed out there were 16 different biomarkers, 16 different proteins which are there, which could be the cause for that led to about 10,000 patients ko jab study kiya. Think for a second. Now, if you have to test some drugs against one of those 16 proteins, which could be the cause, you have to have a method developed, right? अब कैसे पता चलेगा ये protein A है, protein B है, protein C है? So you take that protein out, you clone, you take that protein out. So these techniques. I'm trying to make connection from that was very macro level पे कि ठीक है, non-small cell lung cancer, there are 16 proteins. ठीक है, पता चल गया. अब protein को identify कैसे करें? How do you identify कि कौन सा protein उसमें है? So these techniques, or in order to, let's say for any specific, or c-Myc is one of the protein, for example, if that protein, if you're studying that protein, you have to develop a method which is specific around that specific protein of your interest. So you can create these clones. These techniques basically help you to create those clones, and then in order to develop an assay, so that if you take a drug, if you're developing a drug which is for that specific lung cancer because of c-Myc, for example, so you have to have an assay. When that assay gives you a signal, right? In order to develop an assay, you have to know what sequence is there, and now you take that sequence and create your own assay. Jisme at the end you can clear fluorescent, luminescent, for example. So if the function is that the protein or the drug when interacts with c-Myc, so let's say if that signal in the beginning is 100, whatever that number is, and if a new drug or an entity is blocking that, it goes down to 40, which shows that it causes 60% inhibition, right? 100 hona chahiye tha, 60 ka matlab that drug can cause 60% inhibition. But how do you know what is that inhibition? You need to have an assay. An assay basically is what you clone that cell line or cells. At the end you put a tag. Now in order to clone that, you put the pieces together. First you need to know what its sequence is. आगे किसी चीज़ को बनाना पता चाहिए कि exactly वो है क्या. Then you can in lab you can create that. So that sequencing then helps you to clone that. So fragmentation, these techniques, for example PCR and all that, helps to break down to understand what the protein is, the cause, and then this cloning then helps to create back what you want, so that you can create an assay, and then you can do the testing of thousands of compounds or whatever needs to be done, so you know that for that specific. So इसी तरह से अगर there are 16 proteins, for example, in that case, for each one you can create this, and now you have an assay for each one of those. So cloning is a technique that helps you to create. So biologists in my lab, they do frequently this. If they are working on a specific cell line, they have specific cancer type or any disease type, they look at what is now, because there is so much advance. So many of these proteins, their sequence is known and information is available. So they take that, they create their own vector. And there are companies, you send them that this is the vector, this is the sequence that we need. They create that and they send it back to you, the cell line. You use that cell line now to study your new drugs. Or you can create your own. So, but the technique which is used are the things that we are just talking. To analyze, to do that. So, and again, so there are many other vectors that you can actually create inside. You can create a bacterial artificial chromosomes, you can create yeast artificial. So expression vectors, you can create these vectors, you can design these vectors. But how do you design? You first need to know what they are in order to design that. So these techniques actually help you to do. Okay, we'll stay with Hindi and Punjabi, no Korean. So in order to do that, now that's at a smaller level, but you can actually do at high throughput. You can take a large numbers, you can put, you probably know that when you have 96 well plate, 384 well plate, now there are 1536 well plate. All these, that many wells, and you can actually put your, the things of interest in each of those wells, and you can test those. So you can actually do high throughput. So basically what it does is you can take several genes to identify which gene is being interacted. So let's say, for example, you have a drug, you create a drug in the cell. Your cell are responsive. So this is again going back to see the connection. Yesterday I showed you a slide coming from left or right, phenotypic screening or target based screening, right? So if you start from phenotypic screening, which means a cell, a phenotype, a cell, you test cell whether they survive or they die when you give a drug. So there is a specific cancer cell, let's stay with lung cancer cell line. You take new 1,000 compounds, you test and see some cells, because of the function of those compounds, out of 1,000 compounds, maybe the 20% cell die, in some case 80% cell will die, in case 90, 40, whatever that number is. But those cells, so now you know that those compounds are effective causing the death of the cell. Okay, that's at the micro level. You now want to understand inside the cell which genes are being affected by that. Cell, as I showed you, it's like an ocean. It's a big complex composition of many genes and protein, thousands of genes and proteins, for example. Now, so when you gave a drug and drug is causing the death of that cell, now you need to understand inside the cell which genes are affected. Right? Cell is a macro level. Now you dive deeper into genes. So you can do a high throughput screening where you can take the drug and you can actually do high throughput biology, and that helps you to again to connect what genes or set of genes are being affected. Say for example, so it is possible to do high throughput biology, and again, the functional genomics experiments you can do same way for DNA sequencing, DNA microarray, transcript at the transcript level, structural genomics, metabolomics, and I'll give you a definition of all of these, in the my second lecture today. So basically these information, and based off that there is a lot of information which now bioinformatics, because many of these genes and proteins level information has been already developed, well understood, stored. So you can actually computational way, you have that sequencing and everything is available. You can use bioinformatics in order to make connection make sense, what works, what doesn't work. For example, so I have a student in my lab. She was undergraduate from University of Toronto, cell biology. She was working at Stanford in clinic. Then she did her master's from Hopkins in bioinformatics, and now she's doing PhD in my lab. So when she has to, again, I think after there were a few people and I was just trying to learn, I think generally best is to soak in as much information as possible. So she did cell biology, went to do computational bioinformatics. Now when she is doing her PhD, she's actually very resourceful to other people. So frankly, her own research, when she looks at, she immediately goes to the computational, whatever public domain data is available, and she will try to see how much in the public domain, what type of cancer, what type of disease, and how much is available information. As a matter of fact, she helps in people, those who do not know bioinformatics, by trying to look at and analyze the publicly domain data, and she is co-authors on their work. So knowledge never goes bad. Knowledge is always helpful. So now she is the go-to person in bioinformatics. But bioinformatics, again, because she can do all of these things while she is doing her own. She's working on lung cancer. Her PhD thesis is on lung cancer, and trying to understand and try to develop and test new drugs. But how you can use the known information, so you can have the sequencing information, all of that is available. You can use bioinformatics, which is highly advanced, and that can help you now automation. Things are now more advanced. So I showed you, for example, the simply PCR and restriction enzymes, breaking it down. You can do high throughput screening. You can use, and now that's why these techniques are. And I don't know, here, you probably may know in the biochemistry or biology department, here or somewhere around in other places, in let's say PGI or neighboring institutes, people have these kind of instruments. Very simple. They just simply sequence and they just find out what genes and proteins are working on. But that's basically what they do. That's basically what they do. They look at the genes at the protein level or the at the metabolite level or transcript level by using these techniques, which are now automated. So one can simply have that all that information gathered in order to make better understanding or provide better understanding of what might be happening inside, if the cells of interest, the genes of interest, the proteins of interest, or the tissues of interest in that specific disease. So sequencing, you can sequence the fragment DNA. You can subclone those. Again, at the protein, this could be done at the whole genome. So you can sequence the whole genome. And so I'll give an example of, for example, in my lab, as I mentioned, we design and study a lot of drugs, from natural products to known drugs, to design our own chemical space. Now the question is to understand how in any disease type. So for example, in my lab I have people working on various different types of cancers, basically their area of interest that they have. So when let's say if there is a drug that is at the cellular level is showing very good response, that shows that it actually has an effect. Now you want to understand the gene, so you can do as a very whole genome. So it will give you a whole look at the whole genome and see what genes are upregulated, downregulated, affected, pre-treatment, post-treatment. So you can gather all that information to see if you have a drug, how is that affecting. But you can do the whole genome. So which is again understanding the DNA part, understanding their fragments, understanding what is the sequence, and putting all of that information. It could be simple at a one step at a time. It could be automated. You can do hundreds and thousands together. But you gather all this information by using the simple technique which I showed you earlier. Now things are obviously very advanced. So these can be done in a much faster way. But look at, so larger, you create the vector, as I was telling you, we can create a vector in the lab. So create a large vector. You can source the clone and you can create subclones. You can assemble the subclone to create the cells of your interest, cell lines of the interest that you have. So as a chemist, we talked yesterday about you're designing a drug, and many of you are organic synthetic chemists. You're designing whatever molecules of interest. But now to test those in the biology side. So you can do all of these, the biological side of things, and you can create the cells of interest, the assays of interest, and then at the structure level, at the function level, you can basically start, as I was saying, unrolling एक एक लेयर हटा के बायोलॉजी की, trying to understand at the large level, the smaller level to smaller level. So at the genes level, transcript level, protein level, breaking down to peptide level, breaking down to smaller clones, you can understand one by one, you can unroll so that you can go back to see what is the cause of any protein or how their protein is working. For example, there are proteins, they have various isoforms, and within those isoforms there are only simply 10 base to 14 base difference. They are like 300, 400 base. Just last 10 to 10, that makes the difference. One could be normal and healthy, the other could cause cancer. But there is a very small piece in between that is different. In order to understand, you need to be able to chop entirely to see what is different, how they are modified or changed because of the disease or in the disease. So how do we study that? So these are the techniques which are used to study that, so that it can help in now in many cases there are proteins, because of whatever reason they are modified, they are changed. So you need to first understand what change happened, how that change happened, and where that change happened, which leads to. So for example, there are 2000 around or so transcripts. 4 to 6, they work at the cluster of 4 to 6 transcription factors. How do we understand where the change is, what that change is, and what change actually is important, what change causes the cell function to go haywire and they start multiplying much faster than the normal cell and becomes cancer? At what stage it becomes cancer? Because it doesn't automatically becomes, they are what leads to. So stress, for example, has been one of the environmental factor which is cause. You see tobacco. There are so many ads you see they avoid tobacco. For whatever the regulation level is, कि वो मूवीज़ के अंदर इतना स्मोक करते हुए हीरो को जिसको भी दिखाएंगे नीचे एक छोटा सा लिख देंगे कि टोबैको इज बैड फॉर, तो क्यों दिखा रहे हो? बट दे शो दैट क्योंकि वो अच्छा लगता है, दैट्स ऑन द साइड. बट हाउ डज टोबैको अफेक्ट योर बॉडी? राइट, कुछ तो हुआ ना. बॉडी वॉज नॉट मेड टू के स्मोक एंड चू टोमेटो. सो टोबैको से कुछ तो हुआ. सो वो क्या हुआ? व्हाट चेंज दैट हैपन? Right? So in order to do that, where that change happened, what that change happened. So if the stress level cell ke upar, see what are these? These are quote unquote environmental insults, radiation, your chemical weapon. Now for example, yesterday I gave you an example of the first known the chemotherapy, which started from observation that the mustard gas, when mustard gas, very small molecule, went inside, created blister. कुछ तो उसने change किया ना biology पे. So what that change happened, right? So to be able to understand, as you unroll this more and more, you have to go down to the roots, cause क्या है उसका. But how do we go there? So these are basically techniques that are used to be able to better understand the biology, and based of that to be able to then do the designing of the assays, designing of the models, designing of the techniques that can help you to better test your drugs. In case of chemists trying to design new drugs, design new molecules, design new entities, then be able to test those and without having false negative or false positive. Okay, any question? I don't know what the program is. Is there I think there's a break, right? Half an hour break, and then we meet at 12. 11:45? Okay. So, if you don't have any question, let's take a break and then come back and then I'll go over the omics part. what are the techniques, other techniques which are used to understand the proteins, the genes level. Yeah. Oh. Thank you करें जो सो रहे हैं उन्हें जगा रहे हैं. देखिए मैं मजाक करता हूँ इसलिए क्योंकि लाइटर बना रहे हैं. So you take a break, you come back and we just talk again. By the way, I'm just kidding. Nobody was sleeping. Okay. Don't be bad.
Thank you, sir, for that informative lecture. We have a short tea break and kindly reassemble by 11:45. Welcome back, everyone. We now resume with the lecture.
So, Dr. Singh, when I'm connecting this, okay, molecular biology. So I just mentioned about the few techniques that are used. I'll just go on to now the latest techniques, and I briefly मैंने आपको कहा था पहले introduction part में कि, and again this is just a summary of कि if you want to understand किसी भी चीज़ को, macro biological चीज़ को, वही ना कि आपको पहले समझना पड़ेगा कि वो बनी कैसे है. And these are simple gene level, transcript level, protein level, which is needed because घूम फिर के यही जो bricks जो हैं DNA से लेके ट्रांसक्रिप्ट से लेके जो bricks जो हैं यही बिल्डिंग बनाती हैं, right? तो उसको समझना ज़रूरी है. Now I showed you few techniques. I talked about few techniques which are used frequently. One of the things, how many of you have heard omics, the term omics? Dr. Singh, question. मैं ये पूछना चाह रहा था क्योंकि there is a lot of curiosity among students regarding जो भी उनका कर रहे हैं उससे आगे बेसिस पे वो क्या कर सकते हैं. उन्होंने अगर PhD कर रहे हैं तो PhD के बाद में आगे वो क्या कर सकते हैं, और PhD में जो नहीं है तो PhD उनको कैसे सेलेक्ट करनी चाहिए. So I am happy, ये तो मैं भी करके उसके बाद में I'm happy I'm sit here and ask any question you want, because उसका कारण मैं इसलिए कह रहा हूँ जनरल, क्योंकि many of your questions are common. तो मैं एक को बताऊँगा उसके बजाय I can just give you a session and you can ask me any question that you want about your studies and your career and whatever you want to, और जितना मुझे थोड़ी बहुत समझ है मैं आपको शेयर कर दूँगा. But point being कि same question asked by 5 people, I will be giving the same answer 5 times. So I rather give you question once and everybody can then get that. So I am perfectly fine with that. अगर आपकी इजाज़त है तो. So ऐसा करते हैं, proteomics जो है I'll take another 25-30 minutes or so, जितना भी टाइम लगता है. Just I don't want to rush anything. आप नोट करेंगे, I don't want to rush anything. थोड़ा हो मगर समझ में आए, it's more advantageous कि मैं पूरा रश करके ख़त्म कर दूँ, बोले चला गया ऊपर से सब कुछ. So and at any time, as I was saying yesterday, if you don't think जो मैंने कहा है समझ में आया, just raise your hand, please repeat. I missed it. It's perfectly fine. This is interactive. There's nothing for. When I'm standing here, there is nothing formal. You ask anything. It is ये डॉक्टर सिंह जो एफर्ट कर रहे हैं और मैं वहाँ से आऊँ, ये आपके लिए है, हमारे लिए नहीं है. So make sure you take advantage of जितना भी हमें थोड़ा पता है हम आपको बता सकें. Okay, so what I'm going to do is I'll take few slides. I'll walk over, then we'll turn off the camera, and then we'll have Q&A part.
About that one, no issue.
हाँ, उसके बाद में हम इसको बंद कर देंगे. मतलब ये तो आप रिकॉर्ड करते जाइए, फिर उसके बाद में मतलब Q&A. So that people have very good curiosities, which I like. उनके बड़े क्वेश्चन है, अच्छे क्वेश्चन है. So मेरे जो आंसर मैं देता हूँ सबका बेनिफिट, they all can get the answer. All right, thank you. So we were talking about this machinery. We were talking about understanding this. So how many of you have heard the term omics? आपने प्रोटियोमिक्स तो सुना होगा, जीनोमिक्स तो सुना होगा, राइट? So it is basically a Greek word. इसका origin कहीं ना कहीं तो है ना, which basically mean, as it says here, derived from whole, all, complete. So when you add that om to anything at the gene level, at the protein level, at the transcript level, at the metabolite level, it becomes the whole transcript, whole genomics, whole study of the, you know, at the protein level or the RNA level transcription जो होता है. So these are basically, you know, the things which are very common these days, which are commonly used from the, you know, and then the effect of the not origin, the things that goes through changes, what is known as epigenetic, because of environmental factors. For example, you know, I mentioned earlier, tobacco, smoking, radiation, chemical effect, ये जो भी चीज़ें हैं उसके कारण जो असर होता है एपिजेनेटिक, एपिजीनोम जो चेंज होता है बाद में, उस सब को एट द जीनोम लेवल, स्टडिंग एट द ट्रांसक्रिप्ट लेवल, जो ट्रांसक्रिप्शन होता है, जो चेंज होता है. एंड देयर वाज अ क्वेश्चन ड्यूरिंग द ब्रेक जस्ट नाउ कि कैसे होता है, राइट, फ्रॉम जीन हाउ साइकिल्स यू, आई श्योर यू दैट जो स्टार्टिंग एस फेज, G1 फेज, M फेज, G2 फेज, ये बेसिकली फेजेस है ना, कि आपका जो DNA है, जो आपकी इंफॉर्मेशन है, इट इज बीइंग ट्रांसफर्ड, राइट. हमने खाली उसके नाम दिए हुए हैं बेस्ड ऑन व्हाट वी नो, बट ये सारों को जो है स्टडी करने के लिए किस तरह से करते हैं. सो दीज आर द टेक्निक्स, DNA level, RNA level, at the protein level. So as you go from left to right, you are going from larger size to smaller size, right? From DNA, larger, RNA, smaller, protein, metabolite, and then phenotype. So these are the building blocks. Now what are these things? There are 5 different, as you see here I have techniques. They are broken down, जो अभी जो prevalent, common, which are commonly used technologies. These are basically 5. Genomic, which is the analysis of at the DNA sequencing level, DNA level. That's the genome, genomics. How आपका gene किस DNA, आपके कौन सा DNA है आपको जो genes को बनाते हैं, what is the makeup of that? So you understand that, that's your at the gene level. At transcript, at the RNA level, transcription जो होता है, चेंज जो होता है DNA से RNA, जो ट्रांसक्रिप्शन, जो ट्रांसक्रिप्ट का मेकअप क्या है, that is your transcriptomics, analysis of the transcribed RNA, which is, you know, simultaneous measurement of gene expression values in a cell or in a tissue. So वो जो expression level, and on the left, as you see here, I have in this figure showing कि breakdown कैसे हो रहा है. From here, as you see, it's breaking it down, and then proteomics at the protein level. So DNA, gene, transcription, and then at the proteins, various proteins are from. Then metabolites, identification and quantification of metabolites in a sample. For example, if you have a drug, and yesterday, so I'll tell you how these were useful, because this slide is on the screen. So let me tell you. Metabolite, for example, क्या उसका मतलब है, how does it work? जो drug लिया, there are multiple routes. It could be oral, it could be IV, right? Injection through derm. वो drug को लिया, it has to go through, circulate in the body to go to the site of action, जहाँ पे उसका असर होना चाहिए. अब अगर drug लिया, क्योंकि जाता है, and I was telling yesterday, for example, and I showed you earlier today, that if you take the oral route, आप जो food लेते हैं सीधा आपके stomach को hit करता है, where pH is 2, break down every single thing. अगर pH 2 पे अगर आपने drug लिया, as a chemist think about it, कि if you have a molecule which is not stable at pH 2, so what will happen? आपने drug लिया, और जो drug की जो एक्टिविटी है उस पूरे स्ट्रक्चर पे है कि ड्रग गोज एंड बाइंड्स टू वेरेवर द साइट ऑफ प्रोटीन इज. इफ यू टेक समथिंग एंड यू रिक्वायर कि पूरा का पूरा मॉलिक्यूल एज इज जो लिया है शुड गो एंड इंटरेक्ट विद द प्रोटीन और you know the biomolecule at the site of action. And if you take something which is oral route, and if it breaks down, metabolite uske ban gaye, and those metabolites are not active, will the drug have its drug function? Will it work? What you need is a whole drug. What you need is a whole molecule. वो तो अब टूट गया. It's broken down to small pieces. उसकी drug function is based on the whole molecule function, because जो भी आपने बनाया है, it is interacting on one side of the protein or the other side of the protein in order to make that protein के ऊपर जो भी protein के functions पे असर करने के लिए. But अगर वो टूट गया, मेटाबोलाइट बन गए, वो बना ही नहीं, तो ड्रग तो काम नहीं करेगा. आपने ले तो लिया, बट ड्रग काम नहीं कर रहा. So metabolites को समझना, that's what it says. Metabolites in a sample, whatever that sample is of analysis, उसके टुकड़े होके, क्योंकि क्या है, बॉडी के अंदर enzymes है ना, there are many different type of enzymes. So when anything that goes in, the enzymes are functioning, they are breaking it out, they are chewing the food, and they are doing whatever उनका functions है. If they break it down to and make something which is not the thing that you want, तो काम नहीं करेगा. So how do we study कि उससे मेटाबोलाइट क्या बने? That is what metabolism is. You study the sample कि उसके broken down pieces क्या हैं, what are the metabolites, because that helps you understand. And the reason I use that example is because that this is now you will probably have appreciation of what it takes to bring a drug out. सिर्फ एक डिज़ाइन करके बना दिया means nothing. It is important, but it's nothing. There are 100 other expertise which are needed to, it says it takes a village to raise a child. I don't know if you have heard about the book. कि हर एक को अपना अपना piece contribute करना पड़ता है in order to make the whole thing work, to create the product. So that all of this are very subspecialties within scientific areas. So you have to understand. So that part is known as metabolomics, to understand only the metabolites. And same way then again this micro RNAomics with underlying the control of the transcription. What are the pieces of that RNA वो transcription को जो है control करते हैं. So these now, each one of these have become a full-fledged subspecialty. There are people who do only one of these. There are people who do multiple of this. I think this guy is also doing something. But so that's how. And again, if you look at on the left side, because in order from at the macro level, at the tissue or cell level, to bringing it down, you have to understand all the way to here. क्योंकि जब तक वो नहीं समझ आएगा, सिर्फ आप यहीं पे तक समझते रहेंगे और रूट कॉज ये कैसे चेंज हुए, क्या चेंजेस आए, ड्रग काम नहीं किया, जो मेटाबोलाइट जिस चीज़ के बने वो क्या है, यहीं पे रहे तो उसके रूट तक नहीं पहुँचे, उसकी तह तक, जड़ तक नहीं पहुँचे, तो यू डोंट हैव द होल पजल इन फ्रंट ऑफ यू. जब तक पजल्स को समझ नहीं आएगा, जब तक उसकी आपको क्वेश्चन राइट नहीं पूछा है, आपको पता नहीं चलेगा. तो these are the techniques which are used. And now the things that I talked yesterday and the things which I'm going to talk next, in next few lectures, you will see all these are connecting pieces. Okay, how all these things contribute in trying to understand the biology behind. So it's as actually there was a question asked earlier. कि सर आपकी लैब में सारे कैंसर बायोलॉजिस्ट, somebody asked question. Yes, I'm a trained synthetic chemist. I did my bachelor's in chemistry. I did my master's in applied chemistry and did my PhD in catalysis. And I do only अभी मेरी लैब में very small fraction is chemistry. It's 90% biology. All my PhD students are doing are biologists, cancer biologists. And now you'll appreciate that being chemist doesn't matter which field you belong to. Understanding the fundamental, and that's what I piece by piece, not that I know everything, but that's what my curiosity was, to learn step by step, and then slowly things start to come together, they start to make sense. Okay, DNA is just a larger molecule. RNA is just a larger molecule. And not that you understand every single thing, but at least you begin to understand how it might be working. And again, my interest is drug development, so it helps to understand on both sides. And I must say that I have pretty bright students. कई तो यहाँ से गए, इंडिया से गए हुए हैं, and they are among the best. So as I was telling earlier, the difference is whichever thing you pick, you go to उसके जड़ तक जाके ना उसको choose लो, पूरी तरह से समझ लो उसकी. Take few things. You don't have to be, jack of all trades, but pick few things and master those, and then slowly start building your building on top of that. Anyway, so these are the techniques which are used to understand how this building is made, what is known as the tissue, the organ, the tissue, the cell. So these are basically, few things that I have put here just to show you the appreciation of what it is. And again, many slides इस तरह से इसलिए बनाई हैं क्योंकि you will have all that information. You will have all this information with you. I don't know what their plan is, but all these slides will be shared with them, and so you will have. So मैंने कई जगह पे only bulleted items बनाई हैं, कई जगह पे मैंने details बनाई है. So when you read it, you don't have to go to another reference. You can simply just have most of the information in your hand. So that's basically in medicine. So what we are doing is I'm not doing anything beyond that, just trying to show you how various different knowledge bases are used to understand medicine, therapy, develop drugs, or even to understand what human biology or biology in general is. So these are various techniques, these are various tools. है ना कि आपको वो tools भी होने चाहिए ना, वो औजार भी होने चाहिए करने के लिए. Yeah, that's what basically these are. Any questions? Is it simple? That's why there are no questions, or it is so complicated that there are no questions? Okay, I'll continue. Now that I said that you will have these slides. So again, I will not repeat. I prepared this slide just for you guys, so you have simplified version of what basically genome. ये जो है, it's a systematic study of organisms' entire genome. The human genome, 3 billion base pair. So just imagine 3 billion base pair. उनका permutation combination आप math करके देखिए, right? Or same base pair connected in one way, do one type of function. Same base pair दूसरी तरह से connect किया है, have a different function inside the human body. See, I was telling you yesterday, बनाने वाले की रचना बड़ी मज़ेदार है. And you have to be just Sherlock Holmes of science. Try to investigate it, and before you know, you just overwhelmed with हर चीज़ कितनी well defined है. So it's fun. The reason I'm telling you is that I'm a chemist, but this is so much fun trying to understand. Again, keep your chemistry hat on all the time. When I'm looking at DNA, I'm not looking at DNA. I'm looking at nitrogen and nitrogen, because that is how I am trained. I'm looking at the base pair amino acid. That's how I am trained. But the flip side, it helps. Because a biologist is looking at the whole DNA को समझने की कोशिश कर रहा है, and I'm looking at a smaller pieces of that. So there are always advantages of whatever you have learned. So anyway, again, transcript, study of the entire collection of RNA, ribonucleic acid, and what basically happens. So going back to molecular biology. Again, you saw this from the proteomics study. What you need to do is look at on this side. I have just created this, what it does is you could use. So how is ये होता क्या है? Chromatography, you are familiar with chromatography as a chemist, right? Mass spectrometry, you are familiar with that. So basically now there are techniques, very sophisticated mass specs are available, highly sophisticated mass spec. So as a matter of fact, one of the leading lab in the world is Pacific North National Lab, USK, which is very close to where I am in Oregon. This lab is in east of Seattle. That's probably one of the more highly advanced place for omics के लिए. So basically what they do is they are coming up with the टेक्नोलॉजी को एडवांस करते करते कि the resolution से लेके analytical capability is so much advanced कि आप इसीलिए जीन से लेके RNA से लेके प्रोटीन से लेके मेटाबोलाइट उस लेवल तक आप कर सकते हैं. So we collaborate with them and they collaborate around the world. You can collaborate with them basically online उनके साथ में करके. But this is basically it's a mass spectrometry जो I'm sure आपके डिपार्टमेंट में यहाँ पे भी होगा इंस्ट्रूमेंटेशन पोस्ट में. Mass spectrometry is basically what is used, but it is used to analyze at the DNA level, protein level, DNA level, RNA level, protein level, metabolite level. But simply that technique. But understanding कि आपको जो results आया आप उसको analyze कैसे करना, you have to understand bit about biology कि क्या हम किसी चीज़ का. But technique uses mass spectrometry. That's what is used, simple for analysis. So that is, and you can use in combination with the structural analysis, you can use NMR, you can use X-ray which is used. But again, mass spectrometry is the key. जितने भी कहते हैं हम लोग omics करते हैं किसी भी तरह की, basically they are masters of mass spec techniques. But then they study those biological molecules. So they have well understood that biological molecules at different level, how mass spec, using mass spec, what can be analyzed. So, and this is basically the workflow. You prepare a sample, you separate the protein, select which protein, and you identify those protein. All of this, as you can see here, sample preparation, and the sample could be, again, now you connect this to what I was talking earlier. अगर एक बायोप्सी होती है, एक सैंपल है, पेशेंट सैंपल है, उसकी बायोप्सी हुई, टिश्यू से लिया, जहाँ से भी एक सैंपल को लिया, उसको एक्सट्रैक्ट करके, सॉल्वेंट्स वगैरह से लेके एक्सट्रैक्ट करके, उसमें से आपने प्रोटीन्स को सैंपल प्रिपेयर किया गया, प्रोटीन्स को सेपरेट किया गया, एंड देन प्रोटीन ऑफ इंटरेस्ट that you are, then you run the whole mass spec and see if those proteins are there or not and what is their expression level. And that could be applied. Now for example, if a protein, you take a sample, कोई भी एक sample लिया, चाहे blood sample है, biopsy sample लिया है, saliva sample है, urine sample, कोई भी sample आपने लिया है, उसके अंदर proteins भरे हुए हैं, आपने proteins को लिया. जैसे मैं आपको बताता हूँ कि हम लोग how do we use in my lab this technique. आपने कोई ड्रग बनाना है, ड्रग बनाया आपने एनिमल स्टडीज़ की. सो यू टेक द ड्रग, यू टेक द सैंपल ऑफ अ माइस और सेल लाइन पे जिस पे भी गया है, लेट्स से कैंसर सेल टाइप, व्हाटएवर द टाइप इज, ट्रिपल नेगेटिव ब्रेस्ट कैंसर फॉर एग्जांपल. यू टेक दैट सैंपल, excuse me, you analyze that and see what proteins are overexpressed. Now you treat with your drug the same cell lines. Go. Presumably, if the drug is effective, they did something to that protein. Again, you run the mass spec and see what happened, what protein levels change. So you can identify okay, pre and post treatment what happened, right? In order to understand at the gene level or the protein level, at the transcript level, that's what basically you do. So before treatment of the drug, what your sample was, patients, in this case patient sample, this is a very routine. I don't know, probably यहाँ पे PGI वगैरह जो भी मेडिकल स्कूल वहाँ भी होता है, you take patient sample, you analyze sample, sequencing, NGS, this is next generation sequencing. What is that is done? This is what sequencing, this is what is done. आप उसको analyze कर रहे हैं, तो आपने ट्रीटमेंट से पहले उसको एनालाइज किया तो पता उसमें कौन कौन से हैं मार्कर्स, कौन कौन सी प्रोटीन्स उसमें ओवर एक्सप्रेस्ड हैं. अब आपने उसको ट्रीट किया ड्रग के साथ में, फिर से एनालाइज किया कि कौन से प्रोटीन सप्रेस हुए, और अगर उसका असर होता है देन यू कैन मेक ए कनेक्शन कि इस ड्रग से वो ट्रीट करने से ये प्रोटीन सप्रेस हुए विच वर द कॉज ऑफ द डिजीज. सो नाउ you have some advantage कि आपने आइडेंटिफाई किया. So that's how these techniques, this technique is used. Same way it could be at the gene level. You can, again microarray analysis. Now the techniques are so advanced, it could be on a chip. There are multiple ways to take. एक chip के ऊपर you can have many, एक You think for example एक जो आपके नाखून है 1 centimeter by 1 centimeter उसके अंदर you can have 1000 grooves. You know this microarray जो है microfluidics engineering is very advanced. So you take जो chip analysis, you know, lab on a chip and all जो आपने सुना होगा lab on a chip and analysis on a chip. Basically that's what because each well can have a different type of sample. You can run the sequence. So you, let's say if you have 1000 genes on a chip, and you take a drug, treat with a drug, and you can see कौन सा gene ऊपर नीचे हुआ with that drug. So you know that that drug is affecting that gene or not. Same way it goes with the protein level, transcript level, or whichever. So that's how these techniques, basically that's how these techniques are. And again, as you see here, basically the rest is simply the workflow. But steps are only, you know, so again I have prepared this slide same way to explain to you what protein metabolite, which I already gave you that. So this is basically, in terms of the knowledge, as you see, this is what I was saying just now. If you did the biopsy, and then you extract and you identify उस sample patient sample के अंदर कौन सा genes और proteins हैं, but mass spec and proteomics basically help you to identify. क्योंकि जब identify करेंगे तभी तो treatment उसकी होगी ना कि if you have a drug, as I was back to the example of, for example, 16 different as the cause of lung cancer, किस patient के अंदर कौन सा protein जो है cause है आपको पता चलेगा तभी तो उससे मैचिंग आप ड्रग देंगे. अगर प्रोटीन ए के लिए ड्रग 1 काम करता है तो प्रोटीन बी से लेके बाकी 15 प्रोटीन उसको ड्रग 1 देंगे काम नहीं करेगा. तो पेशेंट बेचारे को आप स्ट्रॉन्ग क्या देते जा रहे हैं, उसका असर हो नहीं रहा. सो दैट्स व्हाट बेसिकली पर्सनलाइज्ड मेडिसिन. यू आइडेंटिफाई कि व्हाट इज द कॉज, विच प्रोटीन इज द कॉज ऑफ द डिजीज, एंड देन यू सी द मैचिंग ड्रग in your kitty, in your toolbox that the physician has, then you try to give. But the techniques, these are the techniques which are used. So if you look at here, right में जो मैंने जो cartoon जिसका है, जो regulatory mechanism, for example, basically what is happening is, you know, from the epigenetic causes, splicing करके protein, you know, post translational protein level पे, gene level पे, transcript level पे, protein level पे. So you keep on analyzing these. But जो technique use, जो omics use होती है, this is what is done. This is a biopsy, same thing could be blood sample, saliva sample, urine sample, but these are the techniques. Basically you run, you identify. So आप ये डब्बा आपने देखे होंगे, ये डब्बे, right? आपने instruments देखे होंगे, but behind the scene that's what they are doing. वो तो ठीक है ना, it's a pretty box created, but inside that's all that is happening. They are just breaking it down to protein and just simply जो भी signal है, signal is captured and that what you see, and then based on that you can identify which protein is present or not. So this is basically, you know, the technique, sampling and techniques which are used. The flow chart is the same. I just wanted to re-emphasize. If you look at this, क्या है sample? It could be any sample, as I mentioned. You create gel, as I was telling you earlier. Use proteomics, again basically mass specs is, and identify the protein. Now this is for the quantitative proteomics. As you see, it helps to understand kitni cheezein, what is the complex mixture of your structure. It helps to understand the spatial arrangement in the microenvironment inside the tissue or inside the cell. It helps you to understand drug mode of action, MOA, mode of action, mechanism of action of a drug. Targets, जो example I told you just now, you would take it before and after treatment. If you look at that, that tells you the drug is working on which genes or not. So it tells you the, you know, drug action. It helps you, you know, in understanding of what signaling pathways, how things are degraded inside the body or the cell, and what disease biomarkers. What are the causes of the disease, the biomarkers, the proteins, the genes जो mark कर सके, identify कर सके. Epigenetic, what changes happen? I gave you example of tobacco, for example, radiation है. Radiation attack, chemical weapons, quote unquote जो हम use जो सुनते हैं, अगर वो chemical weapon, because they are used as a weapon, but that chemical that went and affected, interacted with the biological system, what changes happen? You can learn that again using proteomics and complex system inside the human body. You can see how important and useful tool It is, because it is now the central to any biological, human biology understanding, drug discovery, translational research, and again, the topic of our course is experimental therapeutics. So in order to understand what therapy to create, how therapy works, you need this tool. And this is one of the most advanced tools. So see, it helps basically, experimental therapeutics. This is this is just a summary of what I have been saying for past some time. This is how this omics. Now see how it is connected. आपको पता चले बायोमार्कर क्या है, उसको आइडेंटिफाई करने के लिए could be a blood sample, tissue sample, urine, saliva, जो भी body fluid है. You can use inside, any human part. This is just showing brain का, for example, right? क्या changes आते हैं. You can subtype, identify population, जो मैं कह रहा हूँ, data science, big data, all of that information, all sequencing done for example, again, I use the same example again. If there are 1 million people जिनका data, sequencing data is available, and you have 5 drugs जिनकी treatment the patients have gone through, you can take those 1 million people and you can see before and after treatment क्या हुआ, उनके क्या age group थी, what was their sex or gender, what ethnicity that they belong to. They were Caucasians or Indians or Oriental or black or whatever. उनका जो racial है, so now you can see, you can start forming actually a pattern. Okay, in which of the 5 drugs worked or not worked, what was the common thing in which it worked or didn't work. So you can start making comparison, and to understand all of that, omics is one of the technique which is used. And now you can see as connecting to how the whole experimental therapeutics, how various of these disciplines come together in order to develop a drug. Like I said, next time agar aap crocin or anacin bhi lein, to be thankful to all those scientists who work behind the scene in order to create that. So it's fun. So same way, I'll not blabber more and more on this, but again, same genomic approaches. I mentioned proteomics at the protein level, at the gene level you could do this. So this is basically how our approach. Now think for a second. This is what helps us to appreciate what it takes to create knowledge and make useful application of what we create, and how long it takes. We are in 2025. As I was telling you, these omics, and they are refining again and again as more and more technology, more and more तरीके उसके आते जा रहे हैं कि at a pico, at the micro level, nano level, pico level, उसको करने के लिए आपको instrument भी इतने sensitive होने चाहिए ना कि उसको pick कर सके signals को. So when that engineering aspect is happening, our understanding, it is helping in bio. They are all interconnected. It's helping us to understand better biology, right? But as we understand that, these techniques, for example, sequencing techniques or omics, as I mentioned, coming together to help us to understand कि patient के अंदर है क्या, right? So now you see, with that better understanding, you can so previously there was chemotherapy. Again, using term, for cancer, for example, there was chemotherapy. Then a study showed that you can actually, so chemotherapy में क्या है, paclitaxel or cisplatin or many of these drugs which have been, the basic idea was that drug will attack the cells, cancer cells, it will kill the cancer cells. That's again at a macro level. Inside the cell, as you see, there are many thousands of proteins which are present. So study which came out actually from a senior colleague from my lab, my institute, for the first time ever in the world, showed that you can actually target a specific protein without affecting others and develop the first drug, and then the term targeted therapy started. And the first drug was Gleevec, imatinib, that came out of the lab of Brian Drucker, who is a is a senior faculty in my institute. First time ever in the world in oncology, and now there are 70 specifically targeting TKI, tyrosine kinase inhibitor, BCR-ABL are the proteins for which. So that opened the field of targeted therapy, which is just like 23-24 years ago. It's not too 30 साल से पुरानी नहीं है, वो तब से शुरू हुई. ग्लीवेक आया 22-23 साल पहले. So our understanding of जितना हम समझते आ रहे थे कि है, our understanding of biology is the most important thing. Human health के लिए पहले पता पड़े कि human biology है क्या, किसी की भी health के लिए. And then now, as I showed, now as the technique, for example, next gen sequencing or omics came, you can identify what is the cause of a disease, which protein is the cause, gene or protein is the cause of a disease in the organ. For example, पहले होता था ना कि जो कि head and neck cancer, thyroid cancer, or blood cancer, or triple negative breast cancer, or ovarian cancer, that was named on the organ, tissue level पे. Within the organ, I showed you one example of, for example, in lung cancer, there are known at least there are 16 different proteins. So हर एक का जरूरी नहीं same हो. So now you know कि क्या उसका protein है, which is the cause of that, and you develop and you give to only that. That is now personalized medicine. So see how this, and that was again, now you probably have an appreciation of why I started with the history of. So it takes a long time to develop the knowledge, then to apply that knowledge in a meaningful way to understand the biology. And so here we are, and so there is a paradigm shift now, because now we can understand patient by patient what as best as we can in today's time based on all the knowledge and the techniques that we have. And we have a long way to go. We have a lot to understand biology. इसका मतलब नहीं कि हमको सारा biology जब omics आ गया तो सब कुछ हो जाएगा. That's just one technique. It is better than before in trying. It still doesn't tell you how pura whole biology mechanism, signaling pathways, how they are connected and how they work. We really don't know a whole lot about that. But anyway, as these techniques become more and more available, now we are talking about personalized treatment, right? So from before, like even not too long, 25-30 years ago, before, when you just give a strong toxic drug to kill the cancer cells, now you can actually give better targeted drugs. Now you can give drugs which are relevant to the patient's cause of the protein, the causative protein, the driver of the disease, instead of unnecessarily torturing the patient with all this toxic drug when it is really not going to help. So so there is definitely the treatment patient care has now advanced. Again, these are terminology advanced from before. Still be long way to go, but it is much better than that. So therefore, there is a paradigm shift. One size fits all, which used to be pehle, is not the case. Now you can just actually analyze and you can just treat accordingly. Any question? So what if we take a pause here? I think that's enough for you to digest. And in my folder, and again, if you, the hard drive that he gave yesterday, there was some error. I was trying to copy all my, there were 25 publications, but it was not allowing me to copy that. So maybe I need another one. I can just, copy it and then you can just distribute it whichever way Dr. Singh. But other than that, probably I think there's enough now just for you to digest. And then tomorrow when we meet, we'll talk about pharmacology and toxicology. How, the whole PKPD, what it is, how it works, and how it is helpful in understanding and designing drugs and designing new therapies. Okay, so we have few minutes for you to ask any questions related to what I have said so far, or in general about your education and anything that I can be of any help and I can share with you. Stop. Okay, thanks. So he has given us the signal so we can all go, crack some jokes. Okay. I'll keep this on. I can sit here. Feel free to ask question. Don't hesitate.
Welcome back everyone. Post lunch we are delighted to have Ms. Hina, who will deliver the lecture on topic metal complexes in therapeutics. This lecture will introduce us to promising role of coordination compounds in medicinal chemistry and their mechanism in therapeutic applications.
Thanks, Tanya. Good afternoon, everyone. Myself, Hina, working under supervision of Professor Gurjaspreet Singh. Today, my topic of presentation is metal complexes in therapeutics. So, in the first section, we will discuss about iron. It is a small metal in first transition series in periodic table and has an atomic number 26. It is represented by symbol Fe, which is derived from Latin word ferrum, which is related to its firmness. It is by mass the most common element on Earth, forming much of Earth's outer and inner core. It is the fourth most common element on Earth crust. Then we need to know what does iron do in our body. The iron that we take through food in +3 oxidation state is absorbed in +2 oxidation state in mucosal lining and stored here temporarily as a ferritin complex. From there, it is transferred to various body parts through transferrin, where it may be utilized for synthesis of hemoglobin and myoglobin in RBC and bone marrow respectively. The leftover iron may be stored in macrophages and liver as ferritin complex. So about 70% of iron in body is found in hemoglobin and myoglobin. A large number of enzymes require iron as a cofactor for their functions, as the enzyme involved in oxidative phosphorylation, the metabolic pathway that convert nutrient to energy. Then we need to know what are different functions of iron in our body. Iron support our healthy growth and development. It helps to deliver oxygen to various parts of body. It helps to maintain our energy level and keep the level of oxygen in our muscles. There are two types of iron that is present in food, that is heme iron and the non-heme iron. Heme iron found in red meat, poultry, pork, and it is the most easily absorbable form of iron. The non-heme iron is not as easily absorbed by our body as compared to heme iron, and it includes breakfast cereals fortified by iron, bread, pasta, tofu, bean, dried fruit, and egg. Eating food which enriched with vitamin C, such as tomato, citrus fruit, bell pepper, can also help in the absorption of iron. Then we need to know when are different stages of life where we need to pay extra attention to iron intake. So during infancy and childhood, during adolescence, from the onset of menstruation to menopause, and during pregnancy, also when our diet do not include animal product. Then we will discuss what happens if we don't get enough iron. So when level of iron stored in our body become low, iron deficiency anemia sets in. RBC becomes smaller in size and contain less hemoglobin, as a result of which blood carries less oxygen from lungs throughout the body. The symptoms include iron deficient anemia, including gastrointestinal upset, weakness, tiredness, lack of energy, problems with concentration and memory. Infant and children with iron deficient anemia might develop learning difficulties also. Then we will discuss different stages of life, the effect of iron deficiency. So in pregnant women, during pregnancy, as the amount of blood in women blood increases, so she needs more iron for herself as well as her growing baby. So getting too little iron during pregnancy increases women's risk of iron deficiency, and her infant risk of low birth weight, premature birth, and low level of iron. So, anemia affect on an average 24% of pregnancies and can cause more than just tiredness to the woman and her growing baby. So, it can impact body development of baby. It can slow down the post-delivery recovery, and it can also increase the risk of requiring a blood transfusion. In infants and toddlers, iron deficient anemia can lead to delayed psychological development, social withdrawal, and less ability to pay attention what is happening in their surrounding. By age of 6 to 9 months, full-term infants could become iron deficient unless they eat iron enriched solid food and drink iron fortified formulas. So iron supplementation is very necessary for infants as it prevents anemia, it improves neurodevelopment in iron deficient infant, and it alters metabolism of trace minerals. As we discussed, iron is very important for our body. Also, its excess can cause harmful effects. High dose of iron supplements, especially on empty stomach, can cause an upset of stomach, constipation, nausea, abdominal pain, vomiting, and diarrhea. A large amount of iron may also cause more serious effects, including inflammation of stomach lining and ulcers. High dose of iron can also decrease zinc absorption in our body. Extremely high dose of iron in 100 or 1000 of milligram can cause organ failure, coma, convulsion, and even death. Hemochromatosis is also a disease that is related with excess of iron, and it involves iron absorption and storage in organ like liver, heart, pancreas, leading to potential organ damage. So the health hazards that are related with excess of iron include the amplification of Fenton reaction, which produces reactive oxygen species. The reactive oxygen species so produced may damage DNA, lipid, protein, hamper nutrient absorption, and damage the vital organs. The other health risks that are associated with excess of iron involve disease like cancer, Parkinson's disease, Alzheimer, hepatic fibrosis, and in severe cases may lead to organ failure and even death. So it is very important to monitor the level of iron. WHO has also given the limit of iron in drinking water less than 5.4 micromolar. There are various conventional methods for detection of metal ions that include AAS, ICP-MS, potentiometry, and voltammetry. The main disadvantage of these methods are that they require expensive equipments and trained persons. So we may use naked eye sensor or paper-based sensor for sensing purpose. The main advantage of these methods is that the naked eye sensor provides simple, fast, low cost and high sensitive sensing method, while the paper-based sensor add the portability, disposability and ease of use of the sensor. For this purpose, we have synthesized imine functionalized moiety. So we have taken 2,4-dihydroxybenzaldehyde, dissolved it in DMF using K2CO3 as catalyst. We add propyl bromide to the resulting solution and stir the solution for 24 hours at room temperature. Then the solution was worked up in ice cold water, which result in formation of brown colored PPT. The PPT so obtained were filtered using normal filter paper. Compound 2 so obtained was allowed to react with compound 3, which is amine, and both the compound were dissolved in ethanol, and resulting solution was allowed to reflux for 5 hours at 78 degrees Celsius. Upon completion of reaction, which was monitored by TLC, the ethanol was evaporated under low pressure, resulting in formation of orange colored compound 4. The compound 4 so produced was characterized by 1H, C13, mass and crystallographic method. As it is clear from the 1H NMR, that there is no characteristic aldehyde peak in region from 9 to 10 ppm, showing that it has been completely consumed during the reaction. The peak at 8.95 ppm correspond to the imine proton, and the propyl unit can be represented by peak from 2 to 4 ppm. From the C13 NMR also, we can conclude that as we are not having any peak in region from 180 to 200 ppm, shows that aldehyde been completely consumed during the reaction. And the peak at 156.42 ppm represent the carbon correspond to imine group. The propyl chain can be represented by peak from 56.59 to 78.50 ppm. Then mass spectra show peak at 394.1521, that show the synthesis of our desired product. Crystal of compound 4 were also grown. For this purpose, compound 4 was dissolved in ethanol and was allowed to evaporate under room temperature. Monoclinic crystal system were obtained. The molecule is non-planar with twisted propyl chain. The azo-linked benzene ring were nearly coplanar, and the terminal alkyne show linearity with a bond angle of 180 degree. Figure 2a represents the molecular lattice arrangement of crystal along A axis, that show its zigzag arrangement. Figure 2b represents the arrangement of compound 4 in unit cell, which represent 4 molecules per unit cell. And figure 2c represents the packing of molecule 4 along B axis. Then the synthesized compound was used as naked eye sensor. For this purpose, the compound was dissolved in ACN. The ligand solution exhibited a distinct color transition from pale yellow to vivid orange red upon introduction of iron. No color change was observed with other metal ion, that confirm its high selectivity towards iron. So the unique color change enables naked eye detection of iron without need of any instrumentation. The synthesized compound was also used for paper strip testing. For this purpose, we dissolve the compound in ACN and allow the test strip to soak in it. Then the test strips were allowed to dry at room temperature. Upon introduction of iron solution, there was change in color of strips from yellow to orange. As we can see in figure that at 10 to power minus 1 molar concentration of our probe, there was change in color of strip up to 10 to power minus 3 molar concentration of iron, that show that the threshold concentration was 10 to power minus 3 for iron. Then UV visible response of our compound was also recorded. That was recorded in region 200 to 600 nanometer range. 13 different metal ions were used at 10 to power minus 2 molar concentration. Upon addition of iron, new absorption band emerged at 245, 312, 360 and 496 nanometer. The distinct color change from pale yellow to orange red observed, that supported by appearance of the new peak at 496 nanometer, which was in visible region. Then titration study was also done. For this purpose, iron at 2 into 10 to power minus 5 molar concentration was gradually titrated into 2 ml solution of our compound, which was prepared in which was prepared in ACN with 10 microliter addition per reading. A progressive increase in peak at 496 nanometer was observed, that indicate direct correlation of iron with our synthesized compound. Then LOD was also calculated using linear calibration curve by employing the formula 3 sigma by S, where sigma was standard deviation and S is slope of the curve. The LOD so calculated was 28.71 nanomolar, that confirmed high sensitivity of compound toward iron. Then Bench plot analysis was used to determine the association constant, which was found out to be 2.17 into 10 to power 5 molar inverse. The Joe plot analysis was used to find the stoichiometry between our synthesized compound and the metal ion, which was found out to be 1:1. Then metal interference study confirmed the high selectivity of probe toward iron with no spectral change observed in presence of any other metal ion at 10 to power minus 2 molar concentration. Now in this second section we will discuss about zirconium, that is represented by symbol Zr, is solid at room temperature, is a silvery grey metal with atomic number 40. It is very strong and malleable. The chemical symbol Zr is derived from the name of mineral zircon, which is its primary source. The word zircon itself comes from Arabic word zargan, meaning gold like or as gold. So the name was given due to the golden color of the gemstone zircon. Now we will discuss about application of zirconium and its biocompatibility. Zirconium finds extensive use in a wide array of medical, industrial and nuclear application. Zirconium containing compounds have been used in artificial internal organs at a remarkable rate in recent years. Experimental and clinical studies have supported the broad agreement that zirconium compounds are biocompatible and have low toxicity. So although zirconium compounds are low toxic to humans, the specific risks that are associated with zirconium compounds vary depending upon factors such as its chemical form, its concentration, its duration of exposure and route of exposure to our body. Chronic exposure to high concentration of zirconium compounds can lead to adverse health effects including lung leukonoma, carcinogenesis, pneumonitis, skin irritation and pulmonary gluconoma. NIOSH has determined the exposure limit for zirconium compounds, that was 5 mg per meter cube over 8 hours of workday. The short-term exposure limit was determined to be 10 mg per meter cube. So it is very important to determine the level of zirconium in biological system as well as environment. We can use spectroscopic method. The reason for using these method is that they show high selectivity, sensitivity and adaptability to complex sample matrices. In this study, we have used the spectroscopic method, UV visible spectroscopy and the fluorescence spectroscopy. For this purpose, we have synthesized two structural derivatives of triazole. Both these derivatives show some different characteristics, while some of the properties were common for both of them. For example, the orthotriazole show UV visible and fluorescence response towards zirconium, while both para and orthoisomers show antioxidant activity, anti-cancer activity and molecular docking analysis. Here we have given the synthetic route for probe 8A and 8B, which are structural isomers that are ortho and para triazoles. So for this purpose, we have taken the hydrazine moiety, reacted it with vetraldehyde using ethanol as solvent and refluxing the solution for 5 hours result in formation of Schiff base 3. The resulting Schiff base was allowed to react with dry bromoethane to form the bromo derivative. Then this bromo derivative was allowed to react with sodium azide using DMF as solvent, refluxing the solution for 5 hours at 90 degrees Celsius result in formation of the azide derivative 5. The azide derivative so produced was allowed to react with ortho and para alkyne derivative in presence of copper catalyst and T and THF in 1:1 ratio refluxing the solution at 60 degrees Celsius for 5 hours, resulting in formation of compound 8A and 8B. All the synthesized compounds were characterized by 1H-C13 mass and crystallographic method. So compound 4, which is our bromo derivative, was light yellow solid. The 1H NMR observation of compound 4 show that new ethyl chain give two distinct peak at 3.4 and 3.8 ppm. Each peak correspond to 2 protons. C13 NMR show that bromo-ethyl chain give peak at 28.36 and 51.96 ppm. The mass spectra give peak at 422.94 that also show the synthesis of our bromo derivative. Then azide derivative, which is represented by compound 5, was pale yellow solid and was obtained in 75% yield. The 1H NMR of compound 5 show that there was two distinct peak in region from 3.36 to 3.50 ppm, and each peak correspond to 2 protons. The C13 NMR showed that the azido-ethyl chain showed peak at 48.38 and 49.41 ppm. The peak at 403.24 in mass spectra also confirmed the azide derivative synthesis. Then compound 8A was off-white solid, was obtained in 90% yield, and in 1H NMR, after cyclization of the triazole ring, the CH proton appeared at 7.1 ppm. This OCH2 group that represent the propagyl chain give peak at 5.25 ppm, and aldehyde protons appeared at 10.38 ppm. Aromatic proton give well resolved peak in region from 6.8 to 8 ppm. The C13 NMR show that triazole carbon give peak at 121.33 ppm. Propagyl carbon that was next to oxygen atom give peak at 62.45 ppm, and aldehyde carbon showed peak at 189 ppm. The mass spectra also confirmed the synthesis of our desired product, which is orthoderivative triazole. Compound 8B that give off-white solid and was obtained in 92% yield. The 1H NMR showed that triazole CH proton appeared at 7.11 ppm. OCH2 group of propagyl chain appear at 5.21 ppm, and the aldehyde proton could be seen at 9.88 ppm. Then the C13 NMR show that triazole carbon can be represented by a peak at 124.69 ppm. Propagyl carbon that was next to oxygen appeared at 65.84 ppm, and aldehyde carbon give peak at 190.78 ppm. The mass spectra also confirmed the synthesis of our para derivative. Then the crystals of all these compounds were also obtained. For compound 4 and 5, both of them were dissolved in dry ether and under slow evaporation at ambient temperature result in formation of crystal of both the compounds. Compound 8B was dissolved in ethanol, and same slow evaporation method was applied for crystals. Then preliminary UV visible analysis of 8A and 8B was also done. 8A show peak at 554 nanometer, 311 nanometer, while 8B showed peak at 274 and 311 nanometer. These peak correspond to pi to pi star and n to pi star transitions. That was due to heteroatoms and conjugated pi electron system in these compounds. Then the selectivity by UV visible response of ligand 8A towards zirconium was also recorded. Only zirconium caused a significant change in the spectra. upon 10 microliter addition to our synthesized ligand, the spectral change that was observed was peak at 254 nanometer diminished, the new peak at 275 nanometer appeared, and there was hypochromic shift at 311 nanometer. This may be explained on the basis of HSAB principle and host-guest fit relationship, as the ligand that is a hard base, as it contains nitrogen and oxygen, which are small in size and contain lone pair of electron. These bind with zirconium, which act as hard acid in this case, so they show strong binding. The host-guest fit relationship show that zirconium matches the ligand cavity size and hence enhance the selectivity. Titration study of ligand 8A towards zirconium was also studied. Ligand was taken at 10 to power minus 6 molar concentration, and for zirconium we prepared 10 to power minus 4 molar concentration. There was gradual decrease in peak at 254 nanometer, and the band at 311 nanometer undergo a hypochromic shift. LOD was determined using linear calibration curve, which was found out to be 50 nanomolar. Then association constant was determined using BH plot method, and the association constant was found out to be 0.506 into 10 to power 7 molar inverse. The binding stoichiometry between the synthesized ligand and the zirconium ion was also determined using Joe plot analysis method. Equimolar solution of our synthesized ligand 8A and zirconium was dissolved in methanol, and the volume was taken to be constant at 2 ml per vol, and the inflection point at 0.5 mole, that showed 1:1 binding between both of them. Interference study was used to show the selectivity of ligand towards zirconium and the minimal spectral change from other metal ion that show a strong selective response only in case of zirconium. Fluorescence sensing study was also done with zirconium by ligand 8A. For this purpose, ligand 8A was dissolved in methanol that showed a emission band at 400 nanometer under excitation wavelength at 320 nanometer. There was no significant change with other metal ions, while in case of zirconium, there was a significant fluorescence enhancement. This indicates the specific interaction and high selectivity towards zirconium. Then fluorescence titration of ligand 8A with zirconium was also done. For this, there was a gradual increase in zirconium concentration from 10 microliter to 140 microliter, which was prepared at 10 to power minus 7 molar concentration. In this experiment, the ligand was prepared at 10 to power minus 8 molar concentration. There was weak fluorescence without metal ion, which may be explained on the basis of intramolecular charge transfer and CN isomerization. Upon introduction of metal ion, there was chelation enhanced enfluorescence that affect and reduces the ICT and isomerization, and hence the emission band boosted up. The linear calibration curve was used to find the limit of detection that was found out to be 0.9 nanomolar. Now coming to third section, here we will discuss about nickel element. So nickel, that is represented by symbol Ni in periodic table, has atomic weight 58.693. The chemical symbol Ni for nickel was derived from German term which is Kupfernickel, which translate to false copper or Abel's copper. The German miners use this term to describe a copper-like ore that yield no copper when proceeded, leading to the belief that it was trick played by the devil. So, nickel is an essential trace element for living system and crucial in several industrial applications such as nickel cadmium batteries, electroforming and electroplating. It also has important function in biology also, for example in energy production, metabolic regulation and DNA repair. There are also toxic effect of nickel in our body. For example, its excessive accumulation possesses significant health risk comprising oxidative stress, DNA damage, skin allergies, respiratory illness, central nervous system disorder, and may even lead to cancer. So there is delicate equilibrium between the significance and the toxicity of nickel, emphasizing the crucial need for its sensitivity and selectivity. For this purpose, we have synthesized a compound that contains azo dye. The main purpose of selecting azo dye for this purpose is that azo dye display intense color change that is ideal for visual detection of the analyte. The receptor group in dye binds selectively with metal ion and anion. This binding causes a noticeable color change that is easy and fast to detect. So, it acts as powerful chromogenic sensor for charged analytes and hence useful in industry, biology as well as medical diagnosis. We have also incorporated triazole organosilane in this moiety because it combines the triazole activity with organosilane stability for multifunctional use. Organosilane backbone anchor well with the surface that is ideal for sensor fabrication. Triazole nitrogen rich ring selectively chelate with metal ion and the certain triazole emit fluorescence that change upon metal ion binding. So here the synthetic route for azo dye appended triazole functionalized organosilane. For this purpose we have taken amine moiety and coupled it with phenol to form the azo dye. For this purpose we have taken the amine, dissolved it in water and HCl and allow the coupling reaction with phenol. Upon completion of reaction, the synthesized dye was allowed to react with propyl bromide, resulting in formation of alkyne moiety. This alkyl moiety was allowed to react with azide group to form the triazole ring. So we got triazole functionalized organosilane. All these compounds were characterized by 1H-C13 mass and crystallographic method. As for dye, we can see in 1H NMR that the peak at 4.77, which is a broad peak, represents the proton of OH group. From the mass spectra, we can see that the ketone group, the carbon correspond to the ketone group show peak at 194.37 ppm, while the aromatic carbon atom appeared in range from 106.79 to 160.07 ppm. The mass spectra are showing peak at 340.1339 also confirm the synthesis of the dye. Then for the alkyne, the propyl group show peak at 2.53 and 4.7 ppm, while the aromatic proton can be seen in range from 6.89 to 7.89 ppm. The C13 NMR show that the ketone carbon show peak at 195.63 ppm. The propyl chain can be represented by peak from 55.97 to 77.23 ppm, and the aromatic carbon atom can be represented by peak at 115.05 to 160.36 ppm. In the mass spectra, the peak at 378.14 also confirm the synthesis of the alkyne. Then we come to triazole and capto organosilane. The aromatic region can be represented by peak from 6.82 to 7.79 ppm, and the propyl chain may be represented by peak at 2.24, 2.07 and 2.35 ppm. Each peak corresponds to 2 proton. Then C13 NMR show that the aromatic carbon can be represented in region from 113.94 to 160.20, aromatic carbon represented at 194.59, and the propyl chain give carbon peak at 6.62, 17.40 and 52.81 ppm. The mass spectra show peak at 622.28. Also confirm the synthesis of our final product. Then we also got crystal for our synthesized dye. For this purpose, the dye was dissolved in chloroform and allowed to evaporate the solvent under room temperature, resulting in formation of triclinic crystal system, and the 2 benzene ring in the crystal lie in plane, while the third ring tilted at 120.46 degree out of the plane. Sensing behavior of synthesized organosilane was also recorded by absorption and emission spectra using methanol as solvent. In the UV visible spectra, as we can see, the bands at 2 to 7 nanometer and 378 nanometer in case of our compound. While upon introduction of nickel, there was decrease in peak intensity at 378 nanometer, while new peak appeared at 488 nanometer. For fluorescence spectra, the emission was observed at 457 nanometer under excitation wavelength at 370 nanometer. The strong turn on fluorescence in presence of nickel confirmed the high selectivity toward this metal ion. Then quantitative detection of nickel using azo diorganosilane was also done. So the titration based evaluation was done. There was gradual titration performance using nickel as analyte. UV visible titration protocol show that the probe solution was prepared at 10 to power minus 5 molar concentration, and nickel solution was prepared at 5 into 10 to power minus 5 molar concentration, and there was incremental addition of nickel in 10 microliter slots. From figure we can see that there was absorption change and decrease in peak at 379 nanometer, while increase at 488 nanometer, while fluorescence show that probe show gradual fluorescence enhancement at 457 nanometer. Calibration curve was used to determine the limit of detection. That was found out to be 2.05 into 10 to power minus 7 molar for absorption spectra, and from the fluorescence based LOD was calculated out to be 8.10 into 10 to power minus 8 molar. Association constant was determined using binding interaction method, which involved BH plot approach, and the association constant was calculated out to be 0.36 into 10 raised to power 6 molar inverse. The binding stoichiometry was determined using Joe plot analysis method, and it was found that at mole fraction of 0.5 there was inflection point that show 1:1 stoichiometry between synthesized compound and the metal ion. Interference analysis for nickel detection was also done. The main objective was to evaluate the selectivity of sensor toward nickel in presence of all the other metal ions. We use zirconium, cobalt, barium, cadmium, mercury, etc. The absorption spectra was monitored for change during co-presence of nickel with other metal ions, and there was no significant spectral change in presence of all the metal ions. So sensor exhibited high selectivity and interference detection, free detection toward nickel. Thank you.
Thank you so much, ma'am, for an amazing lecture. We will take a short break of 5 minutes and then resume after that. We have Professor Saraswati, retired Professor, Panjab University, and Dr. Dharamveer from PGI amongst us. I would like to request Professor Gurjaspreet Singh to share a few words about Dr. Dharamveer.
So, good afternoon. Professor Dharamveer is an accomplished associate professor in the Department of ENT at PGI, Chandigarh. With a strong academic background and years of clinical and surgical expertise, Professor Dharamveer has made significant contribution to the field of ENT, particularly in the area of head and neck surgery, endoscopy, sinus surgery, and oncology. He is known for his commitment to patient care, innovative surgical techniques, and academic mentorship. Professor Dharamveer has been actively involved in teaching and postgraduate students and has participated in a number of national and international conferences, workshops, and academic forums. His research interests span a wide spectrum of ENT methods, and he has published extensively in reputed peer-reviewed journals. A respected figure in the medical community, Professor Dharamveer continue to play a pivotal role in advancing both clinical practice and research in ENT and PGI. Now I invite Professor Dharamveer for delivering lecture.
Thank you, sir. So today we are going to talk about the evaluation of middle ear disease. How many of you are aware of ear? Can you raise your hand? कभी सुना है कान के बारे में? किस किसने सुना है? कितने पार्ट्स हैं कान के? 3. सबसे मेन रोल किसका है? मिडल ईयर का. मिडल ईयर इज बेसिकली a bridge. Otherwise main is इनर ईयर. कौन सी नर्व सप्लाई होती है कान से जो सुनते हैं हम लोग? विच नर्व सप्लाई टू द ब्रेन? एनीवन? वेरी गुड. इसके लिए क्लैपिंग. Because this is the main thing through which we are just hearing. We are going to talk about here main few points: physiology and anatomy of the middle ear, identify different external and middle ear pathologies, recognize symptoms and signs of disease. चल रहा है? Is work? Is it working? लेजर इसी से करना पड़ेगा. अच्छा चलो. What are the main functional and anatomy and physiology of the middle ear? Middle ear basically connects external ear to the inner ear. This is a bridge between both external and inner ear. Main function of that is the sound transmission by ossicles. These are the ossicles. When the sound waves activate, it strikes to this tympanic membrane. If you just see this, हम tympanic membrane को और क्या बोलते हैं पंजाबी और हिंदी में? पर्दा. पर्दा. Okay. So it strikes on this. As this push inside, then there is a small teeny bones. We जितनी हमारी body में bones है ना, total. Out of that, these are the main 3 teeny bone we call it. That is, we call it the ossicles. So first, it moves towards the ossicle of that malleus. This is the malleus. Then it strikes like a hammer. Okay. Then it moves towards the incus. Then third one is the stapes. So stapes work like a piston. When this sound waves strike tympanic membrane, it moves these ossicles, and this step is go inside, and it pushes this oval window, which is having a what? Anyone? There is a fluid, perilymph and anolymph fluid are there. These are two watery fluid inside this cochlea. So cochlea के अंदर ये it moves. Then there is a one, it has a basilar membrane. Then basilar membrane moves up and down, and there are hair cells which strike stereocilia, and it moves, and that auditory nerve then start to movement. Then that fibers strike, and we are able to hear in our brain through that auditory nerve. And one is there which is the pressure equalization, which is a main eustachian tube, which equalize. Many times you may have seen that when you are flying, आप फ्लाइट में जाते हो एरोप्लेन में, then आपके कान में कुछ पेन होनी शुरू होती है. इज इट? कभी महसूस करा किसी ने? Why? That is due to the pressure changes. वो प्रेशर चेंज को इक्वलाइज करने के लिए क्या है? ये यूस्टेचियन ट्यूब. It always basically closes, but when it's we are swallowing, then at that time only it's open, and it equalizes middle ear and external ear pressure. Then you have seen third function is that protection by acoustic reflex. Protection by acoustic reflex means you have seen many times when there is a Diwali or some gunshot, you are just trying to close your eyes. Okay, oral reflexes we call it that. But there are in tympanic membrane stapedius muscles which contracts. When the extra sound is there, they just try to control that it should not be ruptured. You should not get the sudden hearing loss. Many times the sudden hearing loss due to this trauma. To prevent that, it helps us. What are the main or most common causes, diseases of the external ear? You can see here, this is a wax. Wax के बारे में तो पता होगा हर एक को. Debris, जो कान में मैल कह देते हैं हम लोग. उसके कारण we get the problem. और external auditory injury, ये canal है, external auditory canal. What happened to this? Many people are using the words, Johnson words, you know that. So when we are using that, this wax go inside, or it hurt your skin from the canal. So that's why also you get injury. You can get edema or trauma with that. Then third one is a congenital ear anomalies. Have you seen any time any child or adult who is not having the pinna? किसी ने देखा आज तक? हमारे पीजे में so many patients are coming nowadays who are having not, ये external ear है ही नहीं उनका, उसको अनोशिया बोलते हैं. अगर फिर ईयर लोब उनके अलग तरह के बैट ईयर के इनके कान, एंड देन यू कैन, कान का बाहर का पार्ट है बट कैनाल इज नॉट देयर. अनोशिया में एब्सेंट ऑफ कैनाल, इसमें माइक्रोशिया में स्मॉल, दिस टाइप ऑफ पिना इज देयर बट कैनाल इज आल्सो देयर. ओके, देन कैप ईयर, क्रिप्टोशिया, एंड ये आपने लास्ट में. देन देयर इज ए बैक्टीरियल इन्फेक्शन. किस में होती है ज़्यादा? जब हम स्विमिंग करते हैं. व्हेन वी आर डूइंग द स्विमिंग, देन यू गेट दैट फंगल, उसको ऑटोमाइकोसिस बोल देते हैं. ऑटो मीन्स ear, mycosis is infection. So this, and maximum problem it gets by the children, swimmers, due to this fungal infection. So we should always swim when we are swimming, we should swim with the earplugs, not without earplugs. Then disease of tympanic membrane, that you can see here the result of negative intratympanic pressure when the eustachian tube is blocked. So इस तरह से उसमें बल्ब, इस तरह की प्रॉब्लम्स, वो जैसे होता है ना दैट हमारे कोई फिंसी हो जाती है, उस तरह से वो बन जाता है जैसा ब्लोसा. और ईयर पेन इज लाइक ए पेन ऑफ डेंटल. कभी किसी को, इफ एनीबडी गेट दैट dental pain, you know that is very severe pain. The same pain you get if you get ear infection. And this basically is only due to some, if you have a cold and cough, whenever you are having the cold and cough, your adenoids and tonsillitis are there, then you get this type of a problem because of this eustachian tube last, which I have shown you, that if that is not working, it's not effective, temperature or is not pressure is not able to maintain, equalize, and the fluid accumulate in the middle ear. Your middle ear is a main cavity in which this pus type fluid is become due to some infection. Then tympanic membrane can rupture. अब कान में जो खासकर लेडीज उन्होंने वो लगाई होती है ना हेयर पेन, दे ट्राई टू कि कान में थोड़ी थोड़ी खारिश होती है तो देन दे गेट द इरिटेशन. एंड इरिटेशन के खारिश तो एक ऐसी चीज़ है, यू नो दैट, कि थोड़ी सी होती है फिर थोड़ी और होती देन इमीडिएटली इट रप्चर योर tympanic membrane, पर्दा. Then if somebody is not having hair pain, then they just try to mastic ले लेते हैं. जॉनसन बर्ड का तो मैंने पहले I have already told you. Then again mostly ruptured by at bus stand के बाहर many times you have seen that the people are sitting, वो कान में ऐसे टांगा हुआ उन्होंने और वो उसका क्लीन करते हैं. हर बंदा जाके, पढ़े लिखे लोग भी, you have seen that they are getting clean from them. Then they also rupture because they don't know the exact measurement of this external auditory canal. Other causes include sudden change in the ear pressure, as I have told, और स्कूल में बच्चों को ज़्यादातर जब टीचर थप्पड़ मार देते हैं, बहुत सारे पेशेंट बच्चे हमारे पास आते हैं मनी टाइम कि उनके कान में टीचर ने थप्पड़ मारा या फादर ने थप्पड़ मारा, मम्मी तो नहीं मारती. So then that is also again get the trauma. Then sudden blast near the ear. अगर आपने देखा दिवाली में many time we are just पटाखे बजाने का बड़ा शौक है यंग जनरेशन को, but उसके कारण we have done the survey in one Diwali 2-3 years back. I know many times every Diwali before one day in the newspaper they publish burn injuries in the firecrackers or the eyes, basically eye trauma, but nobody is talking about that ear trauma. But we have just done this study and we got around 12 patients who got the hearing loss, hearing trauma in the Diwali. Then forceful valsalva. हम basic when your pressure is not able to maintain in the ear, so we just advise this. You have to close your nose and mouth and try to push your ear through the ears. आप कान में से निकालने की कोशिश करो, then you will see that your tympanic membrane comes out. So if you are just you are having the cold and you try to push it more, then also the traumatic rupture of the tympanic membrane. Then pressure by fluid, as I have already told, अगर pressure ज़्यादा बन गया कान के अंदर, mucus बन गया, पस ज़्यादा हो गई, तो then अपने आप ही वो पर्दे को rupture कर देगा. Diving, उसके कारण भी प्रॉब्लम होती है. वाटर स्पोर्ट कई लोग करते हैं, उसमें then forceful stinging and fracture of the temporal bone. Even in the whenever somebody gets a head injury on a roadside trauma, उसके कारण भी the bleeding is coming, कहते हैं ना नाक से blood आएगा या कान से आएगा, that is due to that also trauma. Then perforation of tympanic membrane associated with long standing infections. अगर 3 महीने से ज़्यादा किसी के कान में कोल्ड का 15 दिन से ज़्यादा, नॉट 3 महीने से 15 दिन से ज़्यादा, इफ़ यू गेट दैट इन्फेक्शन इन योर ईयर, देन वो क्रॉनिक इन्फेक्शन हो जाएगी, उसमें कंडक्टिव हेयरिंग लॉस हो जाता है. Immediate referral to higher center should be done in such cases, because if your tympanic membrane rupture, you just get the hearing loss also. Then the classifications of middle ear disease. These are the classification done by them. Infectious may be, non-infectious, congenital, traumatic. In infectious otitis media we call it, that acute or chronic. Acute means 3-4 दिन और 15 दिन से पहले पहले हुआ, अगर 15 दिन के 3 महीने के बाद हुआ तो chronic हो जाएगा. Then this is the mastoid. It goes the infection towards that. अगर mastoid में infection चली गई तो then it can go to the brain also. It's a very serious. So if anybody is getting that pus regularly from the ear, you should get checkup. Non-infectious are those barotrauma, cleistotrauma and ossicular fixation. In ossicular fixation is mostly happen to ladies, maximum in the females, and it is able to confirm at the age when you have around 30 to 35 years, or when the pregnancy is there, then the people come to know that is there any ossicular fixation is there or not. As I have told you that a stapes जो पिस्टन वाला काम करती है उसको कहते हैं, वो स्टेपिस फिक्स हो जाती है ओवल विंडो के ऊपर, तो then it's not moving, so that's why the patient get that hearing loss. Congenital, already told, atresia and ossicular anomalies, and traumatic, tympanic membrane perforation, temporal bone fracture. So these are the pictures through which we can say normal कब होता है, AOM, OME and COM. Then serous otitis media symptoms and signs: reduced hearing, tinnitus, air bubbles behind the tympanic membrane. इस तरह से air bubbles हो जाए. tinnitus का पता आप लोगों को? tinnitus क्या होता है? क्या होता है? जो रात को सोने के टाइम कई बार आपने देखा होगा ना, जो environment में quiet noises पे, the people get the ringing tone. Either many people get that पानी जैसे टप टप करता है उसकी आवाज़ आती है, या घंटियाँ बजती हैं, कहते हैं ना पेशेंट, कई बार कभी सुना होगा आपने, उसको tinnitus बोलते हैं. और tinnitus is really 70 to 80% people are having this problem. इतने पेशेंट आते हैं हमारे पास, मतलब they are really unbearable able to hear those tones regularly. रात को नींद नहीं आती. एक बार मेरे पास पेशेंट आया, she was around 53 year old. मैंने अपने सर से बात करी, उसको profound hearing loss. She was having no hearing in both ear, but फिर भी उसको ये tinnitus की प्रॉब्लम. तो we are not able to diagnose her कि व्हाट इज़ द मेन रीज़न, कि एक बंदे को सुनाई तो देता नहीं, लेकिन फिर टिनाइटस क्यों सुन रहा है? उसको अनडायग्नोस छोड़ना पड़ा, बहुत सारी उसको मेडिसिन दी, सेक्रेटरी वालों ने भी दे दी, न्यूरोलॉजी वालों ने भी दे दी, लेकिन शी वॉज नॉट एबल टू गेट दैट. बट सेडेटिव देते थे फिर सेक्रेटरी वाले, उससे थोड़ा सा उसको आराम मिल जाता. बट सेक्रेटरी की ट्रीटमेंट, यू नो दैट इफ़ यू हैव स्टार्टेड एनी सेक्रेटरी ट्रीटमेंट देन यू हैव अनदर एसोसिएटेड प्रॉब्लम. सो इट्स ऑलवेज बेटर बाबा रामदेव जी, योगा विच द बेस्ट ट्रीटमेंट नाउ डेज, हमारा जो इंडियन कल्चर है उसके लिए. तो यू कैन गेट क्योर विद द बाल्सवा एंड प्राणायाम. ओके, so try to improve with those things is the better treatment. Don't go to the doctors अगर इस तरह से नहीं ठीक होता तो. Then acute otitis media with sudden onset viral infection. Many time आपने सुना होगा, कभी सुना आपने कि रात को पेशेंट सोया हुआ है, सुबह कहता है यार मेरे को तो इस कान से सुनता ही नहीं. So that may be the viral infection. फीवर, ओटैल्जिया, कान में पेन हुई, हेडेक हुई, उसकी वजह से हेयरिंग लॉस हो जाता है. ऑटोस्कोपी, रेड बल्जिंग टिम्पैनिक मेम्ब्रेन दिखाई देती है उसमें, इस तरह से. देन इन चीज़ों का व्हाट्स द मेन ट्रीटमेंट इज एंटीबायोटिक्स ओनली. एंटीबायोटिक्स एंड एनाल्जेसिक्स, पेन रिलीवर, and also anti-inflammatory are there through which you can get them. So ETD, eustachian tube dysfunction, key role in the middle ear. ये देखो यहाँ से जाके ये यहाँ, ये प्रेशर जो हमारा मेंटेन करती है, ये यूस्टेचियन ट्यूब है. और दिस वेंटिलेशन इज़ वेरी वेरी इंपॉर्टेंट. बच्चों में इसका बहुत ज़्यादा इफेक्ट होता है एडिनोइड्स टॉन्सिलाइटिस की वजह से. ये ब्लॉक हो जाती, बहुत छोटी है ये भी, इसीलिए जब इसमें इन्फ्लामेशन हो जाती देन इट क्लोजेस. द चिल्ड्रन आर नॉट एबल टू मेक अ इक्वल प्रेशर. ड्यू टू दैट प्रेशर दे गेट द प्रॉब्लम. मेन कॉजेज एलर्जी, जैसे मैंने बोला पहले, कोल्ड कफ हो गया, इन्फेक्शन हो गई. देन एडिनोइड्स और टॉन्सिलाइटिस, वो स्वेलिंग हो गई टॉन्सिल्स में, एडिनोइड्स में. देन हाइपरट्रॉफी लीड्स टू नेगेटिव प्रेशर, फ्लूइड एक्युमुलेशन, मिडल ईयर में फ्लूइड इकट्ठा हो जाता है. ओके, देन अगेन इसकी evaluation through Valsva. Tympanometry is basically the impedance odometer which make that bridges the, okay, so उसकी measurement के through और endoscopy के through we can check that. Then clinical examination of the ear, inspection, how we do that. आपने देखा होगा कान में डॉक्टर, एक बार एक मेरे फ्रेंड थे, वो जज थे अपने पंजाब हाई एंड हरियाणा हाई कोर्ट के. तो वो मेरे पास ले आए कि इनकी एवेल्युएशन करनी है, टेस्ट करना है. तो जब मैंने उनका टेस्ट करा तो मेरे से ज़रा थोड़ा वो ज़्यादा सेटिस्फाई हो गए, बिकॉज आई हैव स्पेंड मोर टाइम दैन दैट ENT डॉक्टर जो मेन करता है. मेरे को कहने लगे यार मुझे तो ये डॉक्टर जो ENT वाला मेन है, मुझे ये डॉक्टर नहीं लगता. मैंने कहा क्या हुआ सर? कहते यार उसके पास स्टेथोस्कोप नहीं. अब जस्ट इमेजिन दैट अवेयरनेस. एक जज होके उसको इतना नहीं पता कि एक ENT वाले के पास क्या चीज़ होनी चाहिए, और एक मेन डॉक्टर है जो MBBS वाला, उसके पास क्या होना चाहिए. क्या होना चाहिए? कैन यू टेल मी? ENT वाले के पास सिर्फ ये हेड मिरर होता है और ये ऑटोस्कोप होता है, आपको इससे पता चलता है. अब डेंटल वाले को थोड़ी आप मेडिसिन वाला डॉक्टर कह दोगे. आप डेंटल वाले को कहेंगे कि हाँ उसके पास स्टेथोस्कोप है. वो भी सिर्फ एक दिखावे के लिए लगा लेते हैं, लेकिन अब दांत का तो पल्स नहीं देखना ना स्टेथोस्कोप से. सो दैट्स द थिंग कि हमारी अवेयरनेस, व्हाट वी हैव. हमने इतनी किताबें पढ़ ली, इतने जज बन गए, इस लेवल पे आ गए. इफ़ यू डोंट हैव दैट नॉलेज देन कैन यू टेल हमारी एजुकेशन कहाँ है, वेयर वी आर स्टैंडिंग. सो ये छोटी छोटी चीज़ें हैं, वी शुड एबल टू नो दैट. देन पैल्पेशन, ये जो एरिया पेरियर्कुलर इस साइड पे, और ट्रैगस, इसमें टेंडरनेस, इसमें पेन हो जाती है। अगर आप प्रेस करते हो ट्रैगस को, ये जो पार्ट है इसको आप प्रेस करोगे तो पेन होगी आपको। देन मैस्टॉइड टेंडरनेस, ऑलरेडी आई हैव टोल्ड यू कि कान के अगर आप पीछे भी ऐसे हाथ लगा के देखोगे तो आपको पेन होगी इफ़ यू हैव ए इन्फेक्शन इन दैट, और सॉफ्ट हो जाएगा वो। देन ऑटोस्कोपी, थ्रू ऑटोस्कोपी वी जस्ट सी दैट दिस न्यूमेटिक ऑटोस्कोपी मोबाइलिटी, कि जब वो हम ऐसे नाक बंद करके और वो करते हैं तो ये जो ऑटोस्कोप के साथ में वो जो पर्दा है इट मूव्स, तो हमें पता चल जाता है कि TM इज ओके और नॉट। So main, now what type of hearing loss you get if you have these type of infections? So in hearing loss we have three types of hearing loss. One, तो जो whatever he has told about the auditory nerve, जैसे हमारी अगर ऑप्टिक नर्व वीक हो जाती है तो हमें क्या करना पड़ता है, ऐनक लगानी पड़ती है। तो अगर ऑडिटरी नर्व वीक हो गई, उसको SN लॉस बोलते हैं हम लोग, सेंसरी न्यूरल हेयरिंग लॉस, उसके लिए हमें हेयरिंग एड लगानी पड़ती है। उसका यही ट्रीटमेंट है और कोई ट्रीटमेंट ही है। बट इफ इट्स ए 100% profound loss, now the new research is there, like a heart transplant, the same we have a cochlear implant nowadays. अभी जितने भी बच्चे नए भी पैदा होते हैं उनके लिए cochlear implant is available. अगर आपको profound loss 100% deaf mute है बच्चा, तो you can go ahead with this treatment. And this facility, government have so many schemes. If you are poor, from the background of poor, they have some PM fund or social welfare schemes through which they try to help. Otherwise the cost is around 6.5 lakh minimum. So हर patient नहीं afford कर पाता. But if you are working in a government institute, then you can get that benefit from their reimbursement. But it doesn't cover in the Ayushman जो scheme चल रही है. Why? Anybody can tell me? आप लोग young हो, you should know that. Why they have not included in Ayushman? Anyone? Schemes का पता है आपको लोगों को? वैसे जो गवर्नमेंट की स्कीम्स है जिससे पुअर पेशेंट्स को और लोगों को हेल्प मिलती है, डू यू हैव एनी आइडिया? यू शुड हैव, बिकॉज इफ यू आर स्टडिंग इन दिस टाइप ऑफ ए यूनिवर्सिटी, आपके गांव में, आपके घर के आसपास जो कोई बच्चा या बुजुर्ग या किसी को भी कोई प्रॉब्लम इस तरह की हेयरिंग लॉस की है, फ्रॉम वेयर दे कैन गेट द हेल्प, यू शुड ट्राई टू हेल्प दैम. इफ यू हैव अवेयरनेस देन ओनली यू कैन guide them. This is only because it's a cost is they have put the 5 lakh in Ayushman minimum limit, जो maximum limit है 5 lakh. So 5 lakh से cost तो 6.5 lakh की है इसकी. How they can cover that? That's why they have not included in that Ayushman scheme. But heart वाला तो पता है ना, heart वाली हो जाती हैं. जो heart के अभी जो बच्चे जिनका होल होता है कंजेनिटली एबनॉर्मेलिटीज़, उसमें ये भी कंजेनिटल एबनॉर्मेलिटी है. जो हेयरिंग लॉस की मैंने बताई आपको, परफाउंड हेयरिंग लॉस जो है वो कंजेनिटल है, नॉट ट्रॉमेटिक, नॉट दैट. ट्रॉमेटिक में परफाउंड लॉस बहुत कम आता है, बट कंजेनिटल एबनॉर्मेलिटी, जो एबनॉर्मेलिटी कान नहीं बनना जो मैंने बताया, उनमें ये सारा. और उसमें जिसका कान नहीं बना हुआ उसमें इम्प्लांट जो होता है वो 6.8 लाख का है, 80,000 का. उसको बाहा बोलते हैं, बोन एंकर्ड इम्प्लांट होता है वो. सो फॉर प्रोफाउंड लॉस, दिस सेंसरी न्यूरल हेयरिंग लॉस, इस तरह से लॉस आ जाता है. और जब मिडल ईयर में प्रॉब्लम होती है देन कंडक्टिव हेयरिंग लॉस होता है इस तरह से, दिस टाइप. एंड व्हेन द कॉम्बो, हम जाते हैं ना थिएटर में, कॉम्बो की बड़ी आदत है हमें, पॉपकॉर्न मिलेंगे साथ में और कोल्ड ड्रिंक मिलेगी. सो देन वी कॉल इट द मिक्स्ड हेयरिंग लॉस, दैट इज एसएन एंड कंडक्टिव कंपोनेंट बोथ आर हैविंग द प्रॉब्लम. उसकी नर्व में प्रॉब्लम आ जाती है और मिड ईयर में प्रॉब्लम आ जाती है. तो दैट्स व्हाई दे गेट द मिक्स्ड हेयरिंग लॉस, कॉम्बो हेयरिंग लॉस. सो हाउ यू जस्ट इवेल्युएट विद दिस द इम्पीडेंस ऑड. ये जो सेटअप है, दिस अ 2 रूम सेटअप, एनाक्वाइक इसको बोलते हैं हम लोग हेयरिंग टेस्ट, साउंड ट्रीटेड रूम, वी कॉल इट दिस साउंड ट्रीटेड रूम. जब भी हम चेक करते हैं वो एनाक्वाइक वाले नहीं होते. एनाक्वाइक साउंड ट्रीटेड रूम इज दैट विच इज नॉट हैविंग इवन ए जीरो नॉइस, बट जो साउंड ट्रीटेड रूम इज दैट इन विच सम नॉइस लेवल इज देयर, जो हम लोकल अपना जैसे इन हम क्लासरूम वी हैव ए डिफरेंट नॉइस लेवल, in industry we have a different, in Diwali we have a different, in market we have a different noise level. So in this is, we call it the sound treated room. Then this is the impedance measurement through which we just check that if the normal type A audio tympanogram is this, then pressure equal maintain. Here compliances and pressure decapascal units of that, which you just measure with that. If some infection is there, then B type आएगा उसका. If middle ear infection, again उसमें type C आएगा, adenoids or tonsillitis, so pressure कम होगा उसमें. This type of a tympanogram you get. There are two things. One is audiogram, second is audiogram to see that 20 to 8000 hertz, we just check the hearing. But in this only we check the pressure in middle ear, उसमें decibel check करते हैं. How we just get the help with this? Imaging, X-ray, mastoid, outdated, still used in some settings. HRCT, temporal bone, cholesteatoma, ocular erosion, mastoiditis. Then MRI, through which we can check that soft tissues, complications, even pus is there, abscess or facial nerve. Then the medical management. The medical management is only antibiotic or oral topical and decongestant nasal steroids. कई बार पेशेंट कह देते हैं यार कान में मेरे प्रॉब्लम है लेकिन वो नाक में ड्रॉप्स दे रहे हैं डालने के लिए, ये क्या बात हुई? दैट इज ओनली बिकॉज ऑफ यू ड्यू टू यूस्टेशन ट्यूब, क्योंकि जो इन्फेक्शन है वो कंजेशन वहाँ बनी हुई है, वो क्लोज है, उसको खोलने के लिए वी जस्ट गिव दैट टू सी द वेंटिलेशन ऑफ दैट. देन ऑरल टॉयलेट, उसको सक्शन करते हैं जब पस या जैसे फंगस पड़ गई कान में तो उसको क्लीन करना, ऑरल टॉयलेट. देन एनाल्जेसिक्स एंड एंटीपायरेटिक्स. देन सर्जिकल मैनेजमेंट्स आर देयर थ्रू मेरिंगोटमी विद ग्रोमेट इंसर्शन होता है, वो प्रेशर मेंटेन करने के लिए ग्रोमेट इंसर्शन होता है, एक ट्यूब डाल देते हैं टिम्पैनिक मेम्ब्रेन के अंदर तो जिससे वो प्रेशर मेंटेन रहता है. टिम्पैनोप्लास्टी रिपेयर टिम्पैनिक मेम्ब्रेन को करते हैं. ऑसिकुलोप्लास्टी रिकंस्ट्रक्ट करते हैं ऑसिकुलर जो टिनी बोन्स हैं उसको. देन मैस्टोडेक्टमी, फिर पूरा जो मैस्टोडेक्टमी क्लिस्टोमा हो जाता है कान की बैक साइड पे इन्फेक्शन हो जाती है, उसको मैस्टोडेक्टमी बोलते हैं, मैस्टोड की इन्वॉल्वमेंट इन. सो व्हाट आर द मेन पॉइंट्स वी शुड एबल टू नो, जो आपने लर्न करके जाना इससे. दैट इज द मेन रेड फ्लैग साइन जो हैं. परसिस्टेंट फाउल स्मेलिंग डिस्चार्ज, अगर कान में से किसी को रेगुलरली आपके पास बैठा है और मुश्क आ रहा है उसका, तो उसको बहुत ज़रूरी है कि चेक करवाना, क्योंकि वो इन्फेक्शन ब्रेन तक जाती है अगर फाउल स्मेलिंग हो जाए. इवन द कैंसर इज़ देयर नाउअडेज. वो कैंसर किन में होता है? जो डायबिटिक्स पेशेंट हो जाते हैं उन लोगों में बहुत ज़्यादा प्रॉब्लम मैलिग्नेंसी होता है. दैट इज़ वे उनकी तो आप स्मेल पास बैठ ही नहीं सकते अगर इतना उसको हो जाए. देन फेशियल वीकनेस, पोस्ट ऑरिकुलर स्वेलिंग, वर्टाइगो, चक्कर आना. अगर किसी भी को चक्कर कभी आते हों, डोंट गो टू एनीबडी एल्स, गो डायरेक्टली टू द ENT. कोई बताएगा चक्कर ENT के पास क्यों जाना चाहिए? न्यूरोलॉजी के पास सीधा नहीं जाना चाहिए, ऑर्थो वाले के पास सीधा नहीं जाना चाहिए? एनीवन? क्या नाम है उसका? वेस्टिबुलर. ओके, वेरी गुड. तो बेसिकली ईयर में ही वो प्रेशर मतलब फ्लूइड होता है, अगर उसका इम्बैलेंस हो जाता है देन यू गेट दैट प्रॉब्लम. कॉक्लिया के आगे, कॉक्लिया के साथ में वेस्टिबुलर है, तो वेस्टिबुलर पैथोलॉजी इफ देयर इज दैट, तो आपको फिर चक्कर आते हैं. इसीलिए उसका चेकअप इस ऑडियोलॉजिकली बहुत इंपॉर्टेंट है. Then sensory neural hearing loss with middle area pathology. So these are the things, if you have, then immediately urgent requirement to refer to the ENT person. So thank you, thank you, thank you very much. Any question? हाँ. Collect the vibration, collect the sound. आपको शायद याद होगा कि जो डॉग्स हैं दे आर एबल टू हियर मोर देन अस. वो अपनी जब कहते हैं ना कहीं कोई अनहोनी जहाँ होनी होती है, रात को कुत्ते भौंकना और रोना शुरू कर देते हैं. दे जस्ट अपना जो पिन्ना है उस साइड को मोड़ लेते हैं. और कहीं कहीं ह्यूमन बीइंग भी हैं, लड़के भी हैं, लड़कियाँ भी हैं जो अपने कान को पिन्ना को अपने टर्न कर लेते हैं. सो दिस द गिव द मोर कनेक्शन. हाँ जी. एनीथिंग एल्स? एनी अदर क्वेश्चन? फनल तो देखी है ना, फनल क्या काम करती है जब हमने कुछ डालना होता है. समझ आया कुछ बात करने के लिए? क्योंकि आपकी फीडबैक चाहिए मुझे. बिकॉज इतनी बड़ी यूनिवर्सिटी है, पास में है पीजे के, लेकिन आई नो दैट आपने ऑडियोलॉजी सुना है कभी? किस किस बच्चे ने ऑडियोलॉजी सुना है? आपको पता है इसमें स्पेशलाइजेशन होती है ऑडियोलॉजी में. इन आवर पीजे वी हैव ए नाउअडेज, इनिशियली इट वाज ए बीएससी audiology and speech therapy and MSc audiology speech therapy and then PhD. Okay, so now they change the name, that is BASLP, that is a Bachelor Audiology Speech Language Pathology, and then Master Audiology Speech Language Pathology, then PhD in that. So there are main only two institutes, that is one in Bombay, Ali Yavar National Institute Bandra Reclamation, that is the National Institute, and one is the excellent speech and hearing AISH, like AIMS, our PJ. So we call it the All India Institute of Speech and Hearing. So इन दो इंस्टीट्यूट्स हैं इंडिया में मेन जो यहाँ पे ये सारी इसकी ईयर के बारे में, रीहैब के बारे में स्टडी होती है. दे आर द मेन इंस्टीट्यूट्स. थैंक यू वेरी मच.